Hello and welcome back to Secrets After Midnight. Tonight we're going to spend some time with one of the most quietly astonishing transformations in the natural world. Not a caterpillar becoming a butterfly, not a tadpole becoming a frog, but something stranger, and in many ways more unsettling than either of those. We're going to follow an insect that lives two entirely separate lives inside the same body. In its first life, it is a solitary, camouflaged, almost meditative creature, patient, careful, invisible. In its second life, it becomes part of one of the most destructive forces nature has ever assembled. The same animal, the same DNA. Two identities so different that scientists once believed they were looking at two separate species. What changes between those two lives is not a season, not an age, not a predator. It is a single molecule. That molecule and everything it sets in motion is what we're here to explore tonight. So settle in. This is a story that starts very quietly. The desert locust Schistocerca gregaria does not simply grow up and change. It leads two biologically distinct lives, a phenomenon scientists call phase polyphenism. And the boundary between those lives is not written into a calendar or a lifespan. It is written in the nervous system. In its first phase, the solitary phase, the locust is green, reclusive, and behaviorally indistinguishable from the hundreds of other grasshopper species sharing its habitat. It avoids contact. It moves slowly. It feeds with precision. In its second phase, the gregarious phase, it is a different animal in nearly every measurable sense. Different color, different body shape, different brain volume, different behavior, different immune profile, and different relationship to others of its kind. The shift between these two states is not gradual in the way aging is gradual. It can begin within hours of a specific biological trigger. What makes this remarkable is not just that it happens, but that the instruction for both versions of this animal has always been present in the same strand of DNA, waiting patiently for the right conditions to activate one script and silence the other. In its solitary phase, the desert locust wears green with a precision that goes beyond coincidence. Its body color is produced by a combination of two pigment classes, carotenoids, which contribute yellow tones, and bilin proteins, which shift those tones toward the blue-green range, and together they produce a hue calibrated almost exactly to the reflective wavelength of living vegetation. This is not passive coloration. The locust actively modulates the ratio of these pigments based on the light environment it detects around it. Individuals raised on green vegetation in green-lit environments develop deeper, cooler greens. Those raised on yellowing or browning plant matter shift accordingly. The match is never perfect. Nature rarely is, but it is close enough that a resting, solitary locust on a grass stem is for most predators simply not there. Scientists still debate whether this chromatic calibration is primarily driven by diet, by light input through the eyes, or by some combination of both, and the answer may differ between developmental stages. What is certain is that the coloration serves one purpose above all others: to make a small, soft-bodied animal disappear. The solitary locust's ability to tune its own color involves a feedback loop that begins in the compound eye and ends in the cuticle, the hard outer shell that covers the body. When photoreceptors in the eye detect the dominant wavelengths of the locust's surrounding environment, that information travels through the optic lobe and eventually influences the release of hormones, particularly those in the ecdysone family that govern pigment deposition during molting. Each time the locust sheds its old cuticle and grows a new one, the coloration of that new layer reflects the visual data accumulated in the period before the molt. Think of it as a slow, updating photograph. The locust body color is always a slightly delayed portrait of the world it has been sitting inside. A locust that moves to a drastically different background between molts will remain mismatched until its next shed. There are typically 5 molting events before adulthood, which means the system has 5 opportunities to recalibrate. Researchers find this hormonal visual feedback loop genuinely surprising, not because the result is camouflage, but because the mechanism for achieving it is so architecturally precise for an animal that most people walk past without a second glance. The locust's compound eye is not one eye in any sense a human would recognize. It is a mosaic of thousands of individual optical units called ommatidia, each one a self-contained tube with its own lens, its own photoreceptor cluster, and its own narrow field of view. The combined image produced by all of these units covers nearly the full sphere around the animal's head, including a wide arc above and behind, without the locust needing to turn or tilt. Each ommatidium contributes a single pixel of brightness and color to the overall picture, producing a low-resolution but panoramic view that sacrifices fine detail for total spatial awareness. The locust does not see a sharp image the way a hawk does. What it sees is more like a moving mosaic of light, motion, and color, but it sees it everywhere at once. For an insect whose primary survival strategy is remaining still and undetected, this all-around awareness is critical. The moment a shadow crosses the field from above the silhouette of a bird, the arc of a hand, the locust's visual system flags it instantly, and the hind legs are loaded for response before a conscious decision is required. Vision here is less about seeing the world and more about detecting the world's intentions. Embedded within the compound eyes architecture is a capability that goes well beyond simple vision, the ability to detect the polarization pattern of sunlight scattered across the sky. Ordinary light vibrates in all directions. When sunlight passes through the atmosphere, it scatters in ways that create a consistent, the predictable pattern of polarized light, a kind of invisible map drawn across the sky at all times, even on partly cloudy days. The locust's ommatidia contain photoreceptors oriented at different angles, allowing them to sample this polarization pattern and extract directional information from it. The result is a biological compass that functions independently of landmarks and remains accurate even when the sun itself is hidden. This is the same navigational principle used by honeybees and some migratory birds, but its presence in the locust, an insect long studied primarily for its destructive capacity, surprised researchers when it was first described in detail. In the solitary phase, this navigation system operates quietly, helping a lone animal find its way across a landscape while maintaining the spacing and isolation its biology demands. It is, in a sense, the hardware for a journey the solitary locust never intends to take, but that will matter enormously when it does. The locust's hind leg is an engineering achievement, compressed into a few millimeters of biological material. The key to its performance lies not in raw muscle power, but in elastic energy storage, a principle closer to a crossbow than a piston. The large femur of the hind leg contains a specialized muscle called the extensor tibiae, and this muscle, when contracted, does not immediately move the leg. Instead, it loads energy into a semi-rigid region of the leg joint made from a rubber-like protein called rhesilin, mixed with harder cuticle. The leg is held cocked by a second muscle acting as a latch. When the latch releases, the stored elastic energy discharges in a fraction of a second, far faster than muscle alone could generate. The result is a jump that launches the animal skyward in roughly 20 milliseconds. Rhesilin, the protein at the center of this mechanism, has an energy return efficiency of around 97%, meaning almost none of the stored energy is lost to heat. Materials scientists have studied it closely for decades, attempting to replicate its properties in synthetic form for use in robotics and prosthetics. The locust, meanwhile, uses it without awareness every time something moves nearby. A desert locust in its solitary phase weighs roughly 2 grams, about the same as a paperclip. When it jumps, it accelerates its body at forces exceeding 18 times the pull of gravity. To place that in terms that carry weight, if a 180-pound person could match that ratio, they would leave the ground with a takeoff force of more than 3,200 pounds, roughly equivalent to the thrust output of a small aircraft engine applied through 2 legs and released in a heartbeat. The locust achieves this, not through extraordinary muscle mass. Its muscles account for only a fraction of the energy delivered, but through the spring-loading mechanism described in the leg joint. The distinction matters because it means the jump is not metabolically expensive in the way sprinting is for a mammal. The locust can repeat it rapidly, without significant fatigue. In the solitary phase, this jump is primarily defensive, a last resort when camouflage has already failed. The locust does not choose to be conspicuous. It prefers stillness. The jump exists for the moment, stillness is no longer enough, and when it fires, there is almost nothing at human scale that reacts as quickly. Perhaps the most behaviorally distinctive feature of the solitary locust is not anything it does, it is what it consistently refuses to do. It will not tolerate proximity to others of its kind. When two solitary individuals are placed within sensory range of each other, the response is not aggression, not curiosity, and not mating behavior. It is withdrawal. The locust that detects another moves away not urgently but with a steady, deliberate bias in its movement that always increases the distance between them. Scientists describe this as an active spacing behavior, meaning the locust is not simply wandering, it is specifically navigating away from conspicuous members of its own species. The mechanism behind this aversion is multisensory. Visual detection of another locust's shape triggers it. So does the faint chemical signature carried on the cuticle of nearby individuals. Even the ground vibrations produced by another locust's footsteps can be enough to redirect movement. It is a survival strategy. In an environment where every individual is both prey and competitor, spreading out across the landscape maximizes access to food and minimizes exposure to predators who might aggregate where locusts do. The solitary locust, in other words, is not lonely. It is disciplined. The solitary locust is not a machine of consumption. In its isolated phase, it feeds with a selectivity that would surprise anyone familiar only with the swarm version of this animal. Given a choice of plant species, a solitary individual will sample multiple options, assess them through chemoreceptors on its mouthparts and forelegs, and then settle on the combination that most closely meets its current nutritional requirements, particularly its ratio of protein to carbohydrate. This behavior, documented carefully in laboratory feeding trials, is called nutritional self-regulation, and it mirrors strategies seen in far more complex animals. The locust is not eating everything in reach. It is constructing a diet. Researchers have shown that when protein is restricted in one plant, solitary locusts compensate by increasing intake of protein-rich alternatives nearby, and when carbohydrates are scarce, they shift accordingly. The mouthparts themselves are finely structured, serrated mandibles that can strip leaf tissue from a stem without destroying the plant's vascular structure, leaving it alive and capable of regenerating. A single solitary locust feeding across a landscape leaves almost no visible mark. The contrast with what comes later is not merely one of scale, it is a difference in the fundamental relationship between the animal and the plants it touches. Water shapes the solitary locust's world more than almost any other variable. In arid environments across North Africa, the Horn of Africa, and the Arabian Peninsula, these insects have developed a relationship with drought that is counterintuitive. Prolonged dry conditions do not eliminate locust populations, they concentrate them. As vegetation retreats to river margins, oasis edges, and any remaining patch of moisture-retaining soil, solitary locusts follow. They are drawn toward the same shrinking zones of green. At low population densities, this concentration is manageable. Each individual finds moisture and food without crowding its neighbors past the critical threshold. But drought compresses space. It pushes individuals toward the same coordinates, and it is precisely this compression, not malice, not instinct toward destruction, that sets the stage for everything that follows. Scientists have noted that the regions most historically prone to locust outbreaks are not the wettest places in Africa or Asia. They are the places where rainfall is most violently variable, where long dry seasons end in sudden intense precipitation. The drought concentrates. The rain then explodes vegetation, and the concentrated population, now crowded into shared space, crosses a threshold. The individual locust has no awareness of approaching. The solitary locust is ectothermic. It does not generate its own body heat but borrows it from the environment. This dependency, which might seem like a limitation, has produced a surprisingly sophisticated behavioral toolkit for thermal management. In the cool of early morning, a solitary locust will orient its body broadside to the sun, maximizing the surface area exposed to solar radiation. The dark patches present on some individuals' cuticles absorb heat more efficiently than lighter regions, and the locust angles these toward the light source deliberately. As temperatures climb toward midday, the same animal rotates to present only its narrow leading edge to the sun, a posture that dramatically reduces heat absorption. If temperatures continue to rise, It climbs higher on a plant stem where air movement provides convective cooling or shelters in shadow entirely. This behavioral thermoregulation allows the locust to maintain a relatively stable internal temperature across an ambient range that would be lethal without it. Perhaps what's most striking about this process is how little energy it costs. The locust is not running shivering or sweating. It is simply turning, making small postural adjustments that extract or shed heat with the quiet efficiency of a solar panel with legs. The cuticle is the locust's outermost layer, and calling it a shell badly undersells what it actually does. It is simultaneously structural support, waterproofing, membrane sensory interface, color display, and chemical broadcaster, all compressed into a layered material thinner than a sheet of paper. The outermost zone, called the epicuticle, is coated in a thin layer of waxy lipids that dramatically reduce water loss through evaporation. A critical adaptation for an insect living in semi-arid environments where desiccation is a constant threat. Beneath this lies the procuticle, divided into a hard outer layer and a more flexible inner layer, providing the mechanical properties needed for both structural rigidity and joint movement. Embedded throughout are mechanoreceptors, hair-like sensory structures that detect pressure, airflow, and substrate vibration, making the entire surface of the body a distributed sensing organ. The cuticle also carries the locust's chemical identity, a mixture of hydrocarbons on the surface that broadcasts species membership, phase status, and reproductive condition to other locusts nearby. In the solitary phase, this chemical signature signals "leave me alone" to conspecifics. It is in the most literal sense a skin that speaks, and later when the phase shifts, what it says will change entirely. A locust does not hatch as a miniature adult. It emerges from the egg as a first instar nymph, a pale, wingless, soft-bodied creature that bears only a general resemblance to what it will eventually become. Over the course of its juvenile development, it passes through 5 distinct molting stages called instars, each separated by the shedding of the old cuticle and the expansion and hardening of a new one. At each molt, the nymph is temporarily vulnerable. Its new cuticle takes hours to harden, and during that window, it is soft, slow, and conspicuous. The animal typically remains still during this period, choosing sheltered locations and relying entirely on stillness for protection. Between molts, each instar stage brings measurable changes. The body lengthens, the head capsule widens, and the wing buds, initially tiny pads on the thorax, grow progressively more defined. By the 4th instar, the wing buds are clearly visible. By the 5th, they point backward along the abdomen, barely contained, waiting. The final molt transforms the 5th instar nymph into a fully winged adult. The whole process, from egg hatch to adult emergence, takes roughly 5 to 6 weeks under warm conditions, a compressed developmental arc that leaves very little time for error. The 5th instar is the last chapter of the locust's juvenile life, and it carries a particular biological weight. By this stage, the wing buds have grown long enough to fold back along the sides of the abdomen, giving the nymph a compressed, almost tightly packaged appearance, like something waiting to be opened. Inside, the flight muscles are developing rapidly, drawing a disproportionate share of the nymph's nutritional resources. The reproductive organs are also maturing, though not yet active. Endocrine activity, particularly the levels of juvenile hormone, which suppresses adult development, and ecdysone, which drives it, shifts toward a hormonal ratio that signals the final molt is imminent. Scientists still debate the precise environmental cues that determine how long the 5th instar lasts. Temperature clearly accelerates the timeline, but food quality, photoperiod, the daily balance of light and dark hours, and even the presence of other locusts nearby may all contribute to the timing. In the solitary phase, 5th instar nymphs are particularly cautious. They are the most nutritionally valuable they have ever been, and the most physically vulnerable they will be until the molt itself. For many individuals, the 5th instar is the most dangerous weeks of their lives, and the most consequential, because what emerges on the other side will define everything that follows. Most people associate locust noise with the rasping rhythmic calls of late summer evenings, but in its solitary phase, the desert locust is largely silent in the acoustic sense. It does communicate, but not through air. It uses the substrate beneath its feet. By producing micro-vibrations through leg tremulation rapid, low-amplitude oscillations of the hind legs against whatever surface the locust stands on, it transmits signals through plant stems, soil, and leaf tissue that travel to nearby individuals through their own leg sensory receptors. This form of communication, called substrate-borne vibrational signaling, carries information about the sender's presence, size, and in some contexts, reproductive status. The signals are largely inaudible to human ears without specialized equipment. They travel efficiently through rigid plant material but attenuate rapidly in open soil, which means the communication range is short, perhaps a few body lengths. Researchers find this system particularly interesting because it allows locusts to exchange information while remaining effectively invisible to aerial predators that hunt by sound. A locust vibrating a grass stem is broadcasting locally without announcing itself globally, in the solitary phase where survival depends on not being found this quiet. Intimate form of signaling is precisely calibrated to connect when needed and disappear when not. A solitary desert locust, living out its life without the intervention of crowding or transformation, survives for roughly 3 to 5 months as an adult, a span that varies considerably with temperature, food availability, and predation pressure. The egg stage buried in the soil adds another 2 to 4 weeks before hatching, and the nymphal journey through 5 instars occupies another 5 to 6 weeks. The total lifespan from egg to death under favorable solitary conditions falls somewhere between 4 and 7 months. Within that window, a female will lay several egg pods, each containing between 60 and 160 eggs, deposited in moist sandy soil and sealed with a frothy secretion that hardens into a protective plug. She does not guard them. She does not return. The eggs are left to negotiate their own relationship with the environment, a relationship that, as we will see much later, can stretch far beyond the time frame of the mother who laid them. The solitary locust's life is not dramatic by any external measure. It is a sequence of careful decisions about temperature, food spacing, and timing, each one made by an animal that has no awareness of the larger system it participates in. And yet, in aggregate, those individual decisions are quietly building the conditions for something it will never personally witness. The solitary locust is not strictly nocturnal, but it has learned across millions of years of selective pressure that the edges of the day are its safest hours. Dawn and dusk offer a particular combination of conditions that suit its biology, precisely temperatures cool enough to avoid heat stress, light levels low enough to reduce visibility to aerial predators, and enough residual warmth in the substrate to keep its muscles responsive. During the harshest midday hours in arid environments, solitary locusts typically rest in the shade of vegetation oriented to minimize heat gain, metabolically quiet and nearly motionless. Feeding activity peaks in the morning, when plant tissue carries the highest moisture content and the locust's own body temperature is rising toward its optimal functional range. This temporal partitioning different behaviors assigned to different windows of the day is a strategy shared by a remarkable number of desert-adapted animals, from fennec foxes to sidewinder rattlesnakes. The locust arrives at it through a different evolutionary lineage, but the same logic in an environment where heat and predators both kill. The animal that moves at the right time survives. It is worth sitting with that image for a moment, a creature so attuned to the rhythms around it that its entire daily schedule is a negotiation between the sun and the hawk above. In its solitary phase, the desert locust exists within a web of predation that is genuinely diverse. Birds are the most visible predators, particularly starlings, bee-eaters, and various raptor species that can detect and pursue a flushing locust with speed the insect cannot outrun over distance. Reptiles, particularly agamid lizards and chameleons, take solitary locusts from vegetation with the kind of patient strike-and-wait hunting style that the locust's own stillness partially evolved to defeat. Ground-dwelling predators, spiders, mantids, and various beetle species account for significant nymph mortality, particularly during the vulnerable post-molt window when the cuticle has not yet hardened. Amphibians take a seasonal toll near water sources. Even some rodent species under certain conditions will consume locusts opportunistically. The combined pressure of this predator community functions as a population regulator, keeping solitary locust numbers below the density thresholds that would trigger phase transformation. Scientists have noted that in regions where predator diversity has been reduced through habitat loss or persecution, locust baseline populations tend to sit at higher densities closer to the transformation threshold. The predators, in other words, are not merely eating individual locusts. They are collectively and without coordination maintaining the conditions under which a swarm never forms. Beyond the visible predators, the solitary locust contends with a suite of biological threats, too small to see but no less consequential. Entomopathogenic fungi, particularly species in the genera Metahizium and Bovaria, infect locusts through the cuticle, bypassing the gut entirely. A fungal spore that lands on the cuticle surface germinates, penetrates the waxy epicutical layer, and begins consuming the insect from within. Death follows within days to weeks, depending on spore load and temperature. The dead locust often remains clinging to a plant stem positioned by the fungus in a location that maximizes spore dispersal, a manipulation of the host's final behavior that scientists still find unsettling in its efficiency. Parasitic tachinid flies present a different threat. A female fly deposits her larvae on or near a locust, and the larvae burrow into the body cavity, feeding on non-essential tissues while keeping the host alive long enough for their own development. Historians and biologists debate the extent to which these biological controls have shaped locust population dynamics over evolutionary time, whether the locust's immune system adaptations, which are genuinely sophisticated for an insect, evolved primarily in response to fungal pressure, predation, or some combination of both. What is clear is that in the solitary phase, disease and parasitism are constant companions, quiet regulators of an animal that has learned imperfectly to live with them. For a significant portion of entomological history, the solitary desert locust and the gregarious desert locust were recorded, described, and catalogued as entirely separate species. This was not a casual error. The two forms differ so profoundly in color, body shape, wing length, muscle mass, brain volume, and behavior that early naturalists examining preserved specimens had every reasonable justification for treating them as distinct organisms. The gregarious form was well documented, dramatic, abundant, and agriculturally devastating enough to attract serious scientific attention. The solitary form was elusive, cryptically colored, and behaviorally designed to avoid detection, which meant museum collections held relatively few confirmed solitary specimens, and those that existed often showed intermediate characteristics that complicated classification further. It was not until 1921 that the Russian entomologist Boris Uvarov published the work that unified the two forms under a single species and proposed what he called phase theory, the argument that the same animal was producing two radically different phenotypes in response to environmental conditions. The scientific community received this argument with considerable skepticism. The idea that one genome could produce organisms so different without any genetic divergence between them challenged foundational assumptions about what a species was and how development worked. Uvarov was right, but it took decades of subsequent research to demonstrate it with the rigor the claim demanded. With Boris Uvarov's phase theory established, the question that consumed researchers for the following decades was mechanistic. What precisely causes the shift? Early hypotheses centered on nutrition, the idea that food scarcity forced locusts into competitive proximity, and that the resulting stress drove behavioral and physical change. Others proposed temperature, humidity, or some combination of environmental stressors acting on the developing nymph. Each hypothesis had supporting evidence and contradicting evidence in roughly equal measure. The decisive series of experiments came gradually across multiple research groups through careful manipulation of single variables. When crowding was isolated as the independent variable, when food temperature and humidity were held constant, and only the density of individuals was changed, the transformation still occurred. Crowded individuals became gregarious. Isolated individuals remained solitary. The conclusion was unavoidable. The trigger was not resource scarcity. It was not temperature. It was not disease. It was proximity itself, the simple, physical fact of being near others of the same species. This finding restructured the entire framework for understanding locust outbreaks. It meant that a plague was not a response to a shortage, It was a response to an abundance of one another. Scientists still consider this among the most counterintuitive findings in 20th century entomology. When researchers began mapping exactly which part of the locust's body was most sensitive to the crowding stimulus, they found something precise enough to be almost architectural. The inner surface of the hind femur, the large upper segment of the hind leg, carries a dense concentration of mechanoreceptor hairs and associated nerve endings that are disproportionately sensitive to touch compared to other body regions. Stimulating this specific area repeatedly, even on an otherwise isolated locust, can initiate the early stages of behavioral gregarization. Stimulating equivalent areas on the front or middle legs produces a far weaker response. Stimulating the abdomen or antennae in isolation produces weaker responses still. The hind femur's sensitivity makes ecological sense in a crowded, moving group of nymphs marching, jostling, climbing over one another. The hind legs are the most likely body surface to experience repeated contact from neighboring individuals. Nature characteristically placed the trigger exactly where contact would most reliably occur. Some researchers have described this as the locust's crowding detector, a patch of neural tissue that counts, in effect, how many times it has been touched in a given period and begins reporting upward when that count crosses a threshold. The reporting mechanism, as it turned out, runs directly into the brain's serotonin system. The sequence from touch to transformation is not a single event, but a cascade, a chain of biological signals that amplifies as it moves inward from the cuticle surface toward the central nervous system. When the mechanoreceptor hairs on the hind femur are stimulated repeatedly, they generate electrical signals that travel along sensory neurons into the thoracic ganglia, the locust's distributed nervous system hubs, and then forward into the brain. There, the incoming signals trigger the release of neuromodulators that begin altering the activity of circuits governing behavior, movement, and social response. The critical amplification step occurs in the central nervous system, where the arriving signals prompt increased synthesis and release of serotonin. What makes this cascade notable is its sensitivity to repetition rather than intensity. A single, strong stimulus produces little lasting effect. But a moderate stimulus, repeated at short intervals, exactly the pattern produced by the jostling of nymphs in a crowded group, produces a cumulative neurochemical shift that a single event cannot. The locust is not responding to being touched once. It is responding to being touched again and again. And again, a biological counting mechanism that distinguishes transient contact from the sustained social density that warrants a fundamental change in behavioral strategy. It is in miniature a statistical argument made in chemistry. Serotonin is most familiar to a general audience as a molecule associated with mood regulation in mammals, the target of antidepressant medications, the subject of popular neuroscience books, the chemical shorthand for feeling good. In the locust, its role is something far more structural. When serotonin levels in the locust brain rise in response to crowding stimulation, they do not merely adjust the animal's emotional tone. They reorganize the prioritization of entire behavioral systems. Activity levels rise. Feeding rate accelerates. The animal's relationship to movement shifts. Where a solitary locust moves to avoid a gregarizing locust begins to move toward. These changes are not subtle adjustments along a single behavioral dial. They represent a fundamental reordering of what the animal finds aversive and what it finds rewarding. Serotonin in this context is less a mood molecule and more a behavioral operating system, a signal that switches the locust from one pre-written behavioral program to another. The same neurochemical that in humans is linked to social bonding and feelings of belonging is in the locust. The literal molecular switch that converts a committed loner into a creature that cannot stop moving toward others. The parallel is not lost on neuroscientists who study both systems, though they are careful about how far they extend the comparison. One of the most striking aspects of the gregarization process is its velocity. Within 2 hours of sustained hind leg stimulation under experimental conditions, measurable behavioral changes are already detectable. The treated locust begins to show reduced avoidance of other individuals. Within 4 hours, it actively moves toward conspecifics rather than away from them. Within 24 hours, its activity level has increased substantially, its feeding has intensified, and it begins producing the pheromone blend associated with the gregarious phase, effectively broadcasting a chemical invitation to others nearby. The physical changes, color shift, muscle development, wing elongation unfold over the subsequent days and molts, since they require the growth of new tissue. But the behavioral transformation, the part that drives the animal toward others and thus accelerates the density increase, happens faster than almost any researcher initially predicted. This speed matters enormously at the population level. A group of nymphs that reaches the critical density threshold does not spend weeks gradually becoming gregarious. It transitions behaviorally within a day, and in doing so it begins producing the pheromones and behavioral patterns that draw more individuals in, raising the density, further accelerating the transformation in those individuals and creating a self-reinforcing cycle that once initiated moves with a momentum of its own. The evidence linking serotonin to gregarization did not come from observation alone. It came from a series of experiments that were in their design almost elegantly simple. Researchers took isolated, solitary locust animals that had never experienced crowding and injected serotonin directly into their central nervous systems. The results were unambiguous. Within hours, the injected locusts began displaying gregarious behaviors, increased activity, reduced avoidance of conspecifics and movement patterns characteristic of animals that had undergone natural gregarization through crowding. Conversely, when researchers used pharmacological agents to block serotonin receptors in locusts undergoing natural crowding, the behavioral shift was suppressed. The animals remained in crowded conditions but failed to fully transition to gregarious behavior. The receptor blocking experiments were particularly important because they addressed a potential objection that serotonin was merely correlating with gregarization rather than causing it. Blocking the molecule's action interrupted the process. That is the standard of evidence required to move from correlation to causation in neuroscience, and the locust experiments met it. The lead research, published in the journal Science in 2009, was received as a landmark finding, not merely in entomology, but in behavioral neuroscience broadly, because it demonstrated that a single, evolutionarily ancient molecule could serve as a master switch for an entire behavioral identity. Serotonin is the primary neurochemical driver of gregarization, but it does not act entirely alone. Research conducted in the years following the serotonin discovery identified dopamine as a secondary participant in the transformation, playing a role that is distinct from serotonin's but complementary to it. Where serotonin appears to govern the shift in social attraction and aversion, the fundamental reorientation toward or away from other locusts, dopamine seems to modulate the locomotor activation that accompanies gregarization. Gregarious locusts move more. They march. They are restless in a way solitary locusts are not, and this restlessness is partly a dopaminergic effect. Dopamine levels in the locust nervous system rise during gregarization and remain elevated in fully gregarious individuals. Some researchers have proposed that the two molecules divide the transformation's labor. Serotonin rewires the social compass, and dopamine charges the engine that runs on the new heading. The interaction between them is not fully mapped, and scientists still debate the precise sequence, whether serotonin elevation precedes and triggers dopamine changes, or whether both respond simultaneously to a common upstream signal. What is clear is that reducing dopamine signaling in gregarious locusts partially attenuates their hyperactivity without reversing their social attraction, suggesting the two systems, while linked, are not identical in their function or their timing. The locust's antennae are not decorative. In the solitary phase, they function as precision chemical detectors, sampling the air for plant volatiles, moisture gradients, and the faint chemical signatures of other locusts, information used primarily to maintain distance and locate to food. Their sensitivity is high, but their behavioral output is quiet. In the gregarious phase, the same antennae become dramatically more active. Their movement rate increases. Their sampling frequency, the rate at which they sweep through the air to gather olfactory information, rises substantially. The neural pathways connecting antennal receptors to the brain's processing centers show increased baseline activity. The animal is in effect listening harder. This hyperactivation of antennal functions serves the demands of the gregarious life. Navigating within a dense moving crowd requires constant real-time chemical and mechanosensory input. A locust in a swarm needs to track the position and movement of individuals immediately around it, respond to pheromone gradients that indicate direction of swarm movement, and detect the approach of predators through a sensory field that is constantly being disrupted by the presence of millions of neighbors. The antenna in this context is not merely a nose, it is a social instrument, and gregarization tunes it to a frequency that the solitary locust never needed to receive. One of the more subtle but consequential changes that accompanies gregarization is a shift in how locusts respond to the chemical signature of their own species. Solitary individuals, when exposed to the cuticular hydrocarbon blend characteristic of other locusts, show neither strong attraction nor strong aversion. The scent is, chemically speaking, registered but behaviorally ignored, a signal in the environment that carries no particular valence. Gregarious individuals respond to the same chemical blend with measurable attraction. They orient toward it. They follow concentration gradients in the direction of higher chemical density. This shift in olfactory valence, from neutral to attractive, is one of the mechanisms by which early crowd formation becomes self-reinforcing. As a few individuals begin gregarizing and start producing the gregarious chemical profile, that profile becomes an attractant for nearby individuals still in early stages of transformation. Those individuals move closer, contact increases, serotonin rises further, more individuals reach behavioral thresholds. The chemical signal that began as the output of a handful of crowded nymphs becomes within hours to days a beacon drawing in animals from progressively greater distances. Something that begins as a local accident of density becomes, through this olfactory feedback loop, an expanding invitation, and the locust has no mechanism to decline it once the chemistry has shifted far enough. A persistent and understandable assumption about locust swarms is that they begin with scarcity, that hunger drives desperate animals together, and togetherness drives transformation. The experimental record does not support this. When researchers maintain locusts at high density with abundant food, gregarization occurs on the same timeline as in crowded populations with restricted food. When locusts are kept at low density with scarce food, gregarization does not occur, or occurs only partially and slowly. The determining variable isolated across numerous controlled experiments is density of conspecifics, not availability of resources. This finding has a somewhat disturbing implication when applied to natural systems. A region that receives exceptional rainfall, producing abundant vegetation and excellent conditions for locust survival and reproduction, is simultaneously creating the conditions for rapid population growth and the density increase that triggers transformation. The very success of the solitary population, its reproductive abundance in a good year, can generate the crowding that ends the solitary phase entirely. Nature here has constructed a system in which prosperity carries within it the seed of a fundamentally different and more dangerous state. There is no external catastrophe required, only enough individuals close enough together touching each other's hind legs in the warm, resource-rich aftermath of good rain. In the semi-arid ecosystems where desert locusts breed rainfall, events are not evenly distributed across the landscape. They fall in patterns concentrated in certain areas, absent in others, and the vegetation response follows those patterns precisely. A good monsoon season produces green corridors across what was weeks earlier bare desert. Locusts, tracking moisture and vegetation through their sensory systems, converge on these green zones from the surrounding drylands. Populations that were spread thinly across vast territories are drawn toward the same patches of productivity. The concentration happens gradually over days and weeks as individuals from a wide surrounding area migrate toward the best available habitat. From above, and this is now visible in satellite vegetation index data, these green zones light up like targets, and inside those targets locust density climbs. The rainfall does not directly trigger gregarization. What it does is assemble the population in space, bringing together animals that were previously separated by enough distance to maintain solitary behavior. Once assembled, the density threshold does the rest. Scientists tracking the 2019 to 2020 East Africa outbreak were able to observe this process in near real time, using satellite data, watching population concentrations build in vegetation hotspots weeks before. The first swarms were reported on the ground. The rain wrote the invitation. The locusts simply arrived. Gregorization does not require a vast sea of locusts. The density threshold at which behavioral transformation begins to occur is surprisingly low. Research has shown that as few as 4 to 8 nymphs per square meter, a density that would be easy to walk through without noticing, can be sufficient to initiate the early stages of behavioral gregorization in a population of young, highly sensitive juveniles. At this density, contact between individuals occurs frequently enough to provide the repeated hind leg stimulation that drives serotonin elevation. It is a number that resists intuition. Consider a square meter of ground, roughly the area of a large dining table surface. 8 small grasshoppers distributed across that space would be invisible to a casual observer, and yet under the right conditions of nymph age, temperature, and prior exposure history, that density can be the tipping point between a harmless population of cryptic insects and the beginning of a process that ends with billions of animals. Crossing continents. The threshold is not fixed, it varies with developmental stage temperature and the chemical environment, but its general magnitude has been confirmed across multiple studies. The margin between safe and catastrophic in locust population dynamics is thinner than most people would guess. Not all life stages of the locust are equally vulnerable to gregorization. The nymphal stages, particularly the second and third instars, show dramatically higher sensitivity to the crowding trigger than adults. A nymph in its second instar can begin showing measurable behavioral changes after relatively brief periods of crowding. An adult requires more sustained and intense contact to achieve the same degree of behavioral shift. This differential sensitivity has a developmental logic. Nymphs are the stage at which population dynamics are most volatile. Hatching events produce sudden localized concentrations of young animals, and the ability to rapidly assess and respond to that density makes biological sense at the juvenile stage. Adults, by contrast, have already committed to a behavioral profile through the crowding or isolation experienced during development and are more resistant to rapid switching. The age sensitivity gradient also explains why monitoring programs focus particular attention on egg hatching events and early nymphal bands. A population of second instar nymphs at moderate density is far more dangerous in terms of swarm potential than an equal density of adults. The young animal's nervous system is, in a very real sense, still listening for instructions, and crowding is one of the loudest signals it knows how to hear. As individual locusts begin shifting toward the gregarious phase, they begin producing a chemical broadcast that accelerates the process in those around them. The pheromone blend associated with gregarious locusts, a mixture of volatile compounds released from the cuticle and in feces, is detectable by other locusts at remarkably low concentrations. It functions not merely as an identifier, but as a behavioral cue. Nearby individuals exposed to the blend show increased attraction, increased activity, and accelerated behavioral gregorization. The signal once present in an area compounds itself. More gregarious individuals produce more pheromone, creating a higher local concentration, which attracts more individuals, which increases density, which accelerates gregorization further. From the outside, this self-amplifying chemical dynamic looks like a population suddenly making a collective decision. From the inside, to the extent an insect has an inside in any experiential sense, there is no decision at all. Each individual is simply responding to the chemical environment it finds itself in. The collective behavior emerges from a cascade of individual responses, each one locally rational, none of them aware of the aggregate they are producing. Scientists sometimes describe this as an emergent property of chemical signaling, and it is, if you can set aside for a moment how much that phrase understates what it actually produces. The neurochemical changes of gregorization are not purely functional, they are also structural. Sustained elevation of serotonin in the locust brain does not merely alter the activity of existing neural circuits. It promotes the formation of new synaptic connections and the strengthening of existing pathways associated with social processing and locomotor activity. This structural rewiring has been observed in histological studies, comparing the brains of solitary and gregarious individuals. Gregarious brains show measurably higher synaptic density in regions associated with olfactory processing, visual, motion detection, and motor coordination. The mushroom body's structures in the insect brain, associated with learning, memory, and sensory integration, show particularly pronounced changes. Some researchers describe these structural alterations as a form of experience-dependent plasticity, analogous in principle, though not in mechanism, to the synaptic remodeling seen in mammalian brains during learning and development. The comparison should not be overstated, but the fact that a neurochemical triggered by physical contact can produce structural changes in neural architecture, changes that persist and consolidate over time, means that gregarization is not merely a behavioral adjustment. It is a physical remodeling of the organ that generates behavior. The locust is not just acting differently. It is at the level of neural tissue becoming different. The gregarization process does not stop at the brain. It reaches inward all the way to the locust's digestive tract, where a community of microorganisms, bacteria, fungi, and other microbes, collectively called the gut microbiome, undergoes its own transformation in parallel with the animal's behavioral and physical changes. Solitary and gregarious locusts harbor measurably different microbial communities. The gregarious gut is dominated by bacterial species capable of processing a wider range of plant compounds, including some that would be poorly handled by the solitary microbiome. This shift is not a passive consequence of dietary change. It appears to be actively driven in part by changes in the gut's chemical environment produced by the transformation process itself. Some of the volatile compounds produced by gregarious locusts' gut bacteria contribute to the pheromone blend that attracts other locusts, meaning the microbiome is not merely an internal passenger, but an active participant in the fray. Chemical signaling that drives swarm formation. Researchers find this loop genuinely surprising. A shift in gut bacteria helps produce the very chemical signal that draws more locusts together, which increases crowding, which accelerates gregarization, which further shifts the gut microbiome. The locust's transformation, it turns out, is not solely a story about the brain and the nervous system. It is also a story about what lives inside the gut and what that invisible community broadcasts to the world. Modern neuroimaging techniques adapted for use on insects have allowed researchers to observe locust brain activity in ways that were impossible a generation ago. Using a combination of fluorescent markers that indicate neural firing and micro-scale imaging equipment, scientists have compared the brain activity patterns of solitary and gregarious locusts presented with identical visual stimuli, typically the silhouette of another locust moving across a screen. In solitary individuals, the visual stimulus produces a measured, contained response, primarily in the optic lobes, the brain regions directly responsible for processing visual input. In gregarious individuals, the same stimulus produces a response that propagates more broadly, activating regions associated with locomotor preparation, olfactory processing, and what researchers cautiously describe as social attention. The gregarious brain does not merely see another locust. It responds to the sight of one, as though it were an event requiring full system engagement. Some researchers compare this carefully and with appropriate caveats to the difference between noticing a stranger in a room and recognizing a familiar face, the latter producing a cascade of associated processing that the former does not. The locust cannot recognize individuals, but gregarization appears to have made the category of conspecific neurologically significant in a way it simply was not before. The technical term for what happens to the locust phase, polyphenism, is worth examining directly because the concept it describes is stranger and more biologically significant than the word suggests. Polyphenism refers to the capacity of a single genotype to produce multiple discretely different phenotypes in response to environmental conditions. It is distinct from normal variation within a population where different individuals carry different genes and therefore look or behave differently. In polyphenism, the same genetic sequence present in every cell of every individual generates organisms that appear in the locust's case almost incomparably different depending on the environment experienced during development. The genome does not change. The DNA is identical. What changes is the pattern of gene expression, which genes are switched on, which are silenced, and at what developmental stages those switching events occur. This is governed by epigenetic mechanisms, chemical modifications to the DNA and its associated proteins, that alter accessibility without altering sequence. Phase polyphenism in locusts is now considered one of the most dramatic examples of phenotypic plasticity in the animal kingdom, a system in which environment does not merely influence the organism but fundamentally redirects its entire developmental trajectory. The locust does not adapt to its world. Under certain conditions, it produces an entirely different version of itself. The parallels between locust gregarization and human neuroplasticity are genuine enough to be worth stating and limited enough to require immediate qualification. In both systems, experience repeated sensory input produces structural and functional changes in neural tissue. Those changes alter subsequent behavior in ways that reflect the nature of the experience that produced them. In both systems, the changes can persist long after the triggering experience has ended. The locust crowded during a critical developmental window carries neural architecture shaped by that crowding, even if it is subsequently isolated. A human who learns a musical instrument or a second language during childhood carries neural reorganization that persists into adulthood. The principle that experience rewires the brain is shared. What is not shared is mechanism scale, complexity, or experiential depth. The locust's neural changes are driven by a single, well-characterized molecular trigger in a nervous system containing perhaps 1 million neurons. Human neuroplasticity operates across a system of roughly 86 billion neurons through mechanisms of extraordinary complexity, most of them still incompletely understood. Researchers who draw this comparison are careful to say analogous in principle rather than similar in mechanism, a distinction that matters enormously in neuroscience, where superficial resemblances between distant systems have historically produced more confusion than insight. Gregarization does not flip immediately to a permanent state. In its early stages, the process is reversible. A locust that has experienced moderate crowding for a short period can, if isolated, return towards solitary behavioral patterns. But as crowding continues and the structural neural changes described earlier consolidate, a threshold is crossed beyond which isolation no longer restores solitary behavior fully. This point of physiological irreversibility has been studied primarily in nymphs, where the timeline is clearest. After approximately 24 to 48 hours of sustained crowding at sufficient density, the behavioral shift in young nymphs becomes resistant to reversal by isolation alone. The serotonin elevation that began as a transient neurochemical response has by this stage triggered secondary changes in gene expression, synaptic structure, and hormonal profile that persist independently of further crowding. Stimulation. The animal is no longer responsive in the same way to the chemical environment of isolation. It has, in a meaningful biological sense, committed. Some researchers describe this threshold as the locust's developmental decision point, though the word decision implies a deliberative process that is entirely absent. There is no choosing. There is only a cascade that reaches a point from which the path back requires more than simply removing the trigger that started it. Whether a fully gregarious locust can return to a truly solitary behavioral profile is one of the genuinely contested questions in locust biology. The experimental evidence is mixed in ways that have produced sustained disagreement among researchers. Some studies show that gregarious adults, when isolated for extended periods, days to weeks, show partial behavioral reversal. Reduced activity levels decreased attraction to conspecifics, and a gradual shift toward the movement patterns of solitary individuals. Other studies using more rigorous behavioral assays find that the reversal is incomplete, that isolated ex-gregarious locusts retain subtle gregarious tendencies in their movement patterns, sensory responses, and pheromone production that distinguish them from never-crowded solitary individuals. The disagreement partly reflects differences in how reversal is defined and measured. Behavioral reversal and physiological reversal are not the same thing, and a locust can show one without the other. What is broadly agreed upon is that reversal, to whatever degree it occurs, happens more readily in nymphs than in adults, more readily after brief crowding than after sustained crowding, and more readily when isolation is accompanied by the chemical environment of solitary conspecifics rather than simple physical isolation alone. The full answer remains open, which is in its own way one of the more interesting things about it. Laboratory experiments attempting to reverse gregarization have produced results that are useful precisely because they are imperfect. When gregarious locusts are isolated individually and provided with visual, olfactory, and tactile cues associated with the solitary environment, including the chemical profile of solitary phase conspecifics, their behavior shifts measurably over several days. Activity levels decline, the strong attraction to conspecific odors weakens, movement patterns become less directed and more exploratory in the cautious, spacing-oriented way characteristic of solitary individuals. But the reversal plateaus. It does not continue indefinitely toward a fully solitary profile. At some point, the isolated gregarious locust reaches a behavioral equilibrium that is intermediate between the two phases, reduced in its gregariousness, but not restored to its solitary baseline. This intermediate state is itself scientifically interesting because it suggests the two phases are not simply binary switches, but endpoints of a continuous spectrum with stable intermediate positions. It also implies that the conditions required to produce a full solitary phenotype from a gregarious starting point may need to extend across an entire generational cycle, that the children of isolated gregarious individuals raised in full solitary conditions may show more complete solitary characteristics than their parents ever could, regardless of how long those parents were isolated. The transformation of a locust population leaves traces that outlast the individuals who produced them. Crowded locusts deposit feces, shed cuticle fragments, and release volatile compounds into the surrounding environment at rates far exceeding what solitary individuals produce. These materials accumulate in the soil and on vegetation surfaces, creating a localized chemical environment that carries information about past population density. Researchers have found that soil from areas previously occupied by gregarious locust populations retains chemical signals detectable by other locusts for days to weeks after the original population has moved on. Crucially, these residual signals influence the behavior of locusts that subsequently encounter them, both by moderately attracting gregarious individuals and in some experiments by slightly accelerating gregarization in solitary individuals exposed to them. The soil, in effect, holds a memory of crowding, a chemical record of past density that can subtly predispose the next population to move in the same direction. This finding has practical implications for monitoring areas with elevated residual locust chemistry in the soil, represent elevated risk zones for subsequent outbreaks, even when no living locusts are currently visible there. The landscape itself becomes a data point, a slow chemical diary of what passed through it and what that passage left behind. The identification of the serotonin trigger and the density thresholds associated with it has allowed researchers to build more precise, predictive models for locust outbreak risk. Modern climate-based locust forecasting does not simply track rainfall and vegetation. It now incorporates population density estimates, derived from field surveys and remote sensing data, combined with temperature-dependent developmental rate models to calculate the probability that a given population will reach the gregarization threshold within a defined time window. Some models go further, integrating the known sensitivity curves for different nymphal instars and the rainfall-driven vegetation concentration dynamics described earlier, to produce spatially explicit maps of gregarization probability across entire regions. These models are not perfect. Locust biology involves enough stochasticity that precise outbreak prediction remains difficult, but they represent a substantial improvement over earlier approaches that relied primarily on vegetation, rainfall data alone. The incorporation of neurochemical threshold data into large-scale epidemiological modeling is, from a scientific standpoint, a genuinely unusual development. It means that a molecule discovered in a laboratory syringe, injected into a single locust's brain, has become through a chain of subsequent research. A parameter in satellite-scale regional forecasting systems used by international food security agencies. It is worth pausing before moving forward to sit with what the serotonin story actually means, not as entomology, but as a broader observation about the relationship between chemistry and identity. The locust that crowds with its neighbors and crosses a density threshold is not making a choice. It is not sensing danger, calculating odds, or deciding that the time has come to change. A molecule accumulates in its brain. Receptors respond. Gene expression shifts. Behavior changes. The animal that emerges from this process is by nearly every measurable criterion a different animal, and yet it carries the same DNA, the same developmental history, the same body that was days before a creature of solitude, camouflage. What separates those two animals is not genetics. It is not experience in any rich sense. It is a chemical concentration. The threshold between a shy, careful, ecologically harmless grasshopper and the leading edge of one of the most destructive forces in the natural world is at its most fundamental level a matter of how much of one molecule is present in one part of one organ. Scientists are appropriately cautious about drawing philosophical conclusions from biological fact, but the locust does invite a question that is hard to set entirely aside about how thin the line between one kind of being and another might be, and what exactly we mean when we use the word identity. The most visually arresting change of gregarization is the one that happens on the surface, the abandonment of the solitary locust's careful green camouflage in favor of a bold, high-contrast pattern of yellow and black that seems, at first glance, to be the opposite of a survival strategy. For an animal that spent its solitary life perfecting invisibility, the shift toward conspicuous coloration appears almost reckless. The logic, however, is sound, and it reflects a fundamental change in the animal's relationship with predation. In the solitary phase, camouflage works because the locust is alone. A single cryptic insect on a green stem is genuinely difficult to find. In a swarm of millions, camouflage becomes irrelevant. No individual coloration strategy can make one locust disappear inside a cloud of billions. The yellow-black pattern that replaces it is not camouflage. It is a warning. This color combination found across the natural world in wasps, poison dart frogs, and various beetles signals toxicity or unpalatability to visually hunting predators, a strategy called aposematism. Gregarious locusts accumulate plant-derived toxins in their tissues at higher rates than solitary individuals, and the coloration honestly advertises this. The locust has not abandoned survival strategy. It has exchanged one for a completely different one, appropriate to its new social reality. The shift from green to yellow-black is not painted on from outside. It is produced from within through a reorganization of the same pigment-producing systems that generated the solitary green. The yellow component of the gregarious coloration comes primarily from an increase in carotenoid deposition, the same pigment class present in green form, but now expressed at higher concentrations and without the blue-green modifying proteins that shifted it toward foliage wavelengths in the solitary phase. The black patterning is produced by melanin, a pigment synthesized through a biochemical pathway that is upregulated during gregarization by hormonal signals associated with the transformation. Specifically, changes in juvenile hormone levels and ecdysone ratios during the molting process alter the activity of enzymes in the melanin synthesis pathway, producing heavier melanin deposition in regions of the cuticle that were previously lightly pigmented. The result is a color pattern that consolidates across successive molts, each instar showing more pronounced yellow-black contrast than the previous one, until by the adult stage, the gregarious coloration is fully expressed. The transition is not instantaneous. A locust in the early stages of gregarization may show intermediate coloration patches of yellow emerging against a background still carrying traces of green before the full pattern stabilizes. These intermediate individuals are, in a sense, visible proof of a transformation still in progress. Aposematism, the use of conspicuous coloration to warn predators of unpalatability or toxicity, is one of the more elegant solutions evolution has produced to the problem of being edible. It works because predators learn. A bird that eats a brightly colored insect and experiences an unpleasant or toxic response will subsequently avoid similar coloration, and that learned avoidance generalizes to other individuals with the same pattern. For the system to function, the signal must be honest. The conspicuous animal must actually be unpalatable, or the warning coloration loses its value. Gregarious locusts maintain this honesty. They sequester plant-derived phenolic compounds and other secondary metabolites in their tissues at rates significantly higher than solitary individuals, a consequence of their broader dietary range and faster gut throughput. These compounds make gregarious locusts genuinely less palatable to many predators. Field observations have documented birds selectively avoiding gregarious phase locusts while readily consuming solitary phase individuals of the same species. The aposematic system also benefits from the swarm's scale. A predator learning to avoid yellow-black locusts in a swarm of millions is effectively learning to leave the entire swarm alone, a collective protection that no individual camouflage strategy could provide. The coloration is simultaneously a personal warning label and a property of the group. The wings of gregarious locusts are not simply larger versions of solitary wings. They differ in shape, proportion, and functional architecture in ways that reflect the completely different demands of gregarious life. Solitary locusts use their wings primarily for short escape flights, reactive high-power bursts that carry the animal away from a threat and into cover. Their wings are proportioned accordingly, adequate for explosive short-distance flight, not optimized for sustained travel. Gregarious wings are longer relative to body size, with a higher aspect ratio, a shape associated in aerodynamics with efficient, sustained flight at the expense of maximum acceleration. The wing venation pattern also differs subtly, with gregarious wings showing structural reinforcement in regions that bear the highest mechanical stress during prolonged flapping. The flight muscles attached to these wings are larger in proportion to body mass, and they are metabolically configured for aerobic endurance rather than anaerobic burst performance. These differences do not develop gradually over many generations of natural selection in the conventional sense. They emerge within the single lifetime of the individual animal, guided by the same developmental shift that produced the color change and the behavioral reorganization. The same serotonin-initiated cascade that rewired the brain also instructed the body to build different wings, a detail that still strikes many researchers as one of the more astonishing aspects of what is already an astonishing process. Accompanying the wing changes is a substantial reorganization of the locust's muscular system, one that reflects the shift from a sedentary explosive response lifestyle to one built around sustained movement across large distances. The flight muscles of gregarious locusts, the large indirect flight muscles that occupy much of the thorax, increase in mass relative to body weight compared to their solitary counterparts. More significantly, their metabolic profile changes. Solitary flight muscles contain higher proportions of fast-twitch fiber types, optimized for rapid, powerful contractions over short durations. Gregarious flight muscles shift towards slow-twitch configurations, fibers that generate less peak force but can sustain contraction for far longer before fatiguing. This is the same fundamental distinction seen in the difference between a sprinter's leg muscles and a marathon runner's, a comparison that translates across a vast evolutionary distance. Because the underlying physics of muscle performance do not change between insects and mammals. The locust does not choose to build endurance muscles. The developmental program initiated by gregarization builds them automatically, guided by hormonal signals and altered patterns of gene expression during growth. The animal that emerges from the gregarious transformation has a body that is specifically architected for a journey it has never taken, and in many cases will not survive. Of all the physical changes that accompany gregarization, perhaps the most conceptually striking is the one that occurs inside the skull, or rather inside the hard exoskeletal head capsule that serves the locust in the skull's place. The mushroom body's paired brain structures found in insects that are associated with olfactory learning, spatial memory, and the integration of sensory information across modalities are measurably larger in gregarious locusts than in solitary individuals of equivalent age and body size. The increase in volume is not trivial. Studies have documented mushroom body expansions of 30% or more in fully gregarious individuals compared to solitary controls. This growth reflects an increase in the number and density of neural connections within these structures, rather than simply an increase in cell number, a distinction that matters because it suggests enhanced processing capacity rather than mere structural inflation. The mushroom bodies in insects generally are thought to support behavioral flexibility, the ability to modify responses based on experience and context. A gregarious locust navigating within a swarm of billions, responding to rapidly changing pheromone gradients, visual motion fields, and mechanosensory input from neighbors, faces far more complex information integration demands than a solitary individual tracking a single path across empty terrain. The brain, instructed by the same developmental cascade that built longer wings and endurance muscles, grows to meet those demands. The enlarged mushroom bodies of gregarious locusts are not simply larger, they appear to function differently in ways relevant to the specific challenges of swarm life. Research examining the learning and memory performance of solitary versus gregarious locusts in controlled behavioral tasks has found that gregarious individuals show enhanced performance in tasks requiring the integration of spatial and olfactory cues, precisely the combination most relevant to navigating within a dense, chemically complex swarm. Gregarious locusts learn odor location associations more rapidly and retain them more accurately over longer intervals. They also show greater behavioral flexibility, the ability to update a learned response when the conditions that originally produced it have changed. These cognitive differences are modest by the standards of vertebrate cognition, and researchers are careful not to overinterpret them. The locust is not becoming intelligent in any rich sense, but within the narrow functional domain of swarm navigation, tracking pheromone gradients, maintaining orientation relative to swarm movement, responding to rapidly shifting local density, the enhanced mushroom body function provides a genuine performance advantage. It is a brain precisely sized for the problem it needs to solve. No larger, no smaller, which is in its own quiet way a description of good engineering regardless of the system producing it. Migration across continents requires fuel, and the gregarious locust's body anticipates this demand in advance of the journey. During the final nymphal instars and the early adult stage, gregarious individuals accumulate fat reserves at substantially higher rates than solitary locusts of equivalent nutritional intake. The fat body, a diffuse organ distributed through the locust's abdomen that functions as its primary energy storage tissue, grows proportionally larger, and the composition of stored lipids shifts toward forms optimized for sustained aerobic metabolism during flight. Simultaneously, the enzymes responsible for mobilizing these fat reserves, releasing stored lipids into the circulatory system for delivery to flight muscles, are upregulated in gregarious individuals, providing faster access to energy during sustained flight bouts. The result is an animal that carries more fuel, accesses it more efficiently, and burns it in a muscular system configured to use it over long distances. Think of it as the biological equivalent of a long-haul aircraft configured for transoceanic flight, extra fuel capacity, efficient engines, and a flight profile optimized for distance rather than speed. The locust does not plan its journey, but its body, remodeled by gregarization, is prepared for one in ways its solitary self never was and never needed to be. The gregarious locust's digestive system is rebuilt for a different relationship with food, one characterized by volume, speed, and breadth, rather than the selective precision of the solitary phase. The gut of a gregarious individual is measurably longer than that of a solitary counterpart, providing greater surface area for nutrient absorption. The rate at which food moves through this extended gut is also higher. Gregarious locusts show faster gut transit times, meaning material is processed and passed more quickly. This faster throughput enables the animal to consume and extract nutrition from larger quantities of plant material per unit of time, supporting both the energetic demands of sustained flight and the rapid fat accumulation described previously. Perhaps more consequentially, the gregarious gut's enzymatic profile is broader. Solitary locusts are selective feeders whose digestive enzymes are calibrated to the plant species they preferentially consume. Gregarious locusts produce a wider range of digestive enzymes, enabling them to extract nutrition from plant species that solitary individuals would largely ignore or find difficult to process. This dietary breadth is what makes swarms so comprehensively destructive. They do not specialize. They consume across the available botanical spectrum with an efficiency the solitary locust's more refined digestive system simply cannot match. The expansion of dietary breadth in gregarious locusts is not merely a behavioral choice. It is underwritten by specific changes in gut biochemistry. Studies examining digestive enzyme activity in solitary versus gregarious individuals have found significantly elevated levels of cellulase activity in gregarious guts enzymes capable of breaking down cellulose, the structural carbohydrate that forms plant cell walls and constitutes a major component of most vegetation. Solitary locusts, as selective feeders targeting nutritionally dense plant tissues, have less need for high cellulase activity. They consume the soft, nutrient-rich parts of preferred species rather than processing large volumes of structural plant material. Gregarious locusts, consuming rapidly and broadly, require the ability to extract nutrition from tougher, more structurally complex plant material. Protease activity, the suite of enzymes that break down plant proteins, is also elevated in gregarious individuals, reflecting the higher protein demands of rapidly developing flight muscle tissue. These enzymatic changes are partly driven by the altered gut microbiome described earlier. The bacterial community that shifts during gregarization contributes its own enzymatic capacity to the digestive process. The locust and its gut microbes are in this sense co-transforming two biological systems simultaneously, reorganizing themselves toward the same functional end, each change reinforcing and enabling the other. The cuticle of a gregarious locust is not simply the same shell worn by its solitary counterpart at a later developmental stage. It is a materially different structure, denser, harder, and more resistant to mechanical damage in ways that reflect the physical demands of life inside a swarm. In a moving mass of millions of individuals, each locust is in constant contact with neighbors, jostling, being climbed upon, pressed against others during roosting, and subjected to wing turbulence from individuals flying immediately adjacent. A cuticle optimized for a solitary life of minimal contact would sustain cumulative mechanical damage under these conditions. The gregarious cuticle addresses this through increased sclerotization, a chemical process in which the protein matrix of the cuticle is cross-linked and stiffened by quinone compounds, producing a harder, more impact-resistant surface. The degree of sclerotization in gregarious individuals is measurably higher than in solitary counterparts, particularly in the thoracic regions that bear the greatest mechanical load during flight and contact. The epicuticular wax layer also thickens, improving resistance to abrasion. Researchers examining gregarious cuticle under electron microscopy describe a material that has been, in effect, upgraded for a more physically demanding environment, the biological equivalent of switching from standard to reinforced materials without changing the underlying blueprint, simply because the conditions of use have changed so dramatically. Reproduction in the gregarious phase operates under a different strategic logic than in the solitary phase. Solitary females living in stable, low-density environments invest in producing larger numbers of eggs across multiple laying events distributed through their adult lifespan. Gregarious females shift toward a different allocation, fewer eggs per laying event, but eggs that are larger, better provisioned with yolk and enclosed in a more robust protective pod. The egg pods produced by gregarious females are buried at higher densities, often in the same locations as other gregarious females, producing aggregated egg fields in the soil, and are structurally reinforced to withstand the physical compression of adjacent pods and the disturbance of overlying soil movement. The eggs themselves carry higher lipid reserves, providing the developing embryo with more energy to draw on during the early stages of development. Perhaps most significantly, gregarious egg pods are sealed with a foamy secretion that hardens into a particularly effective moisture retention barrier, extending the range of soil moisture conditions under which successful hatching can occur. Fewer eggs, individually stronger. The gregarious female is not reducing her reproductive investment, she is concentrating it, trading quantity for individual robustness in offspring that will hatch into an already crowded world and need every advantage the eggs' provisions can supply. When gregarious eggs hatch, what emerges is already fundamentally different in behavior from a solitary hatchling. First instar gregarious nymphs do not disperse. They aggregate. Within hours of hatching, they begin moving in the same direction as their neighbors, not because any individual is leading, and not because they can perceive the overall group, but because each nymph is tracking and matching the movement of the individuals immediately around it. This local alignment, repeated across thousands of individuals, produces the coordinated directional marching that characterizes nymphal locust bands. These bands, sometimes called hopper bands, since the wingless nymphs move by walking and jumping, can extend across areas of several square kilometers and contain millions of individuals moving with a coherent directionality that appears from above almost purposeful. The march is sustained through the daylight hours and pauses at night when the nymphs cluster together for warmth. Each morning, the band reforms and continues in broadly the same direction as the previous day. Navigation draws on the same polarized light detection described in the solitary stage. But now that hardware, which the solitary locust used to maintain spacing and isolation, is being used to maintain collective directional coherence. The same tool, a completely different application. That reuse of existing sensory infrastructure for a radically new social function is in retrospect one of the more elegant aspects of a transformation that has already accumulated quite a few. The sustained directional consistency of marching hopper bands has puzzled and fascinated researchers for decades. Individual nymphs have no map, no memory of the destination, and no awareness of the band's overall size or shape. Yet bands maintain coherent headings across days of travel, shifting direction gradually in response to vegetation gradients, topography and wind, but rarely reversing, rarely fragmenting into independent subgroups, and rarely stalling without environmental cause. The mechanism behind this collective directionality involves several interacting factors. Polarized light provides a stable celestial reference that individual nymphs use to maintain consistent heading. Pheromone trails deposited by the leading individuals create a chemical track that followers reinforce and extend. Visual alignment, each nymph matching its movement direction to the average motion of visible neighbors, produces a positive feedback between individual alignment and group coherence. And the threat of cannibalism, which is real and documented in marching bands, provides a powerful incentive to keep moving. A nymph that stops is at risk of being consumed by those immediately behind it. Scientists debate which of these factors is dominant under different field conditions, and the honest answer is probably that the relative contributions vary with terrain, temperature, and band density. What is not debated is the outcome: a coordinated march that covers kilometers per day, driven by an algorithm so simple that each participant requires almost no cognitive complexity to execute it. The vibrational communication system, described in the solitary phase, does not disappear during gregarization. It is repurposed. Gregarious locusts produce stridulatory signals, sounds generated by rubbing body parts together at higher rates and with different spectral characteristics than solitary individuals. Where solitary vibrational signals were quiet, short range, and functionally oriented toward maintaining spacing, gregarious stridulation is louder, more frequent, and associated with coordinating movement and signaling alarm within the group. The hind femur, already identified as the primary site of the gregarization trigger, also carries the file and scraper mechanism responsible for stridulation in many locust species. The row of pegs on the inner face of the femur that, when rasped against the edge of the forewing, produces the characteristic locust sound. In gregarious individuals, this mechanism is used more actively, and the signals it produces travel through both substrate and air. Large marching bands produce a collective acoustic environment, a combined stridulatory output from thousands of individuals that is audible to humans at some distance. People living near active hopper bands describe it as a dry, continuous rasping like wind through dead grass but emanating from the ground. It is not a sound that carries obvious menace in isolation. In context, knowing what produces it, it carries something else entirely. Life inside a swarm is immunologically demanding in ways that solitary existence is not. A locust in a crowded group is in constant physical contact with millions of conspecifics, each carrying its own microbiome, its own pathogen load, and its own potential for transmitting disease. Pathogen transmission rates in dense populations are, by the basic mathematics of contact frequency, vastly higher than in dispersed solitary populations. The gregarious locust's immune system has adapted to this environment through a combination of elevated baseline immune activity and altered immune prioritization. Gregarious individuals show higher circulating levels of antimicrobial peptides, small proteins that disable bacterial cell walls, and more rapid immune activation in response to pathogen challenge. They also show greater tolerance of bacterial loads that would trigger fatal immune responses in solitary individuals. The mechanism behind this tolerance is not fully understood, but researchers believe it involves a recalibration of the threshold at which the immune system launches a full inflammatory response. A recalibration that reduces the metabolic cost of constant immune surveillance in a high-exposure environment. The solitary locust's immune system is calibrated for a low-pathogen world. The gregarious locust's is calibrated for a crowd, and the difference, under natural conditions, is the difference between an immune system appropriate to its context and one that is chronically overwhelmed by it. As gregarious locusts accumulate plant-derived toxins in their tissues, the compounds that underwrite the warning coloration described earlier, they face a challenge that their solitary counterparts never encounter the toxins they sequester can be harmful to themselves if they accumulate beyond manageable concentrations. Gregarious individuals have evolved a partial solution to this self-toxicity problem through upregulation of detoxification enzymes in the fat body and gut tissue enzymes capable of chemically modifying sequestered compounds into less harmful derivatives while retaining enough of the original chemical character to maintain the aposematic signal. This detoxification capacity is measurably higher in gregarious individuals than in solitary ones. It represents a biochemical investment. The production of detoxification machinery has metabolic costs that is only worthwhile when the animal is accumulating toxins in the first place, which only occurs at the dietary breadth and intake rates characteristic of the gregarious phase. Researchers find this a particularly clean example of coevolution between a morphological strategy, warning coloration, requiring toxin accumulation, and a biochemical detoxification capacity to manage the accumulated toxins. The system is not perfectly efficient. Heavily gregarious individuals do show some evidence of toxin-related metabolic stress under laboratory conditions, but it is functional enough to have persisted across the evolutionary history of Evilage, the species. Every aspect of the gregarious locust's transformation points in the same metabolic direction: higher. Oxygen consumption rates in gregarious individuals are substantially elevated compared to solitary counterparts at rest, reflecting the energetic costs of heightened immune activity, larger and more active muscles, expanded brain tissue, and the constant locomotor activity that characterizes gregarious behavior. During flight, the metabolic rate of a gregarious locust is among the highest recorded for any insect of comparable body size. The mitochondria, the cellular organelles responsible for converting fuel to usable energy, are more abundant in gregarious flight muscle tissue, and their individual efficiency is higher. The circulatory system, which in insects is an open hemolymph system rather than a closed vascular network, moves at a higher pumping rate in gregarious individuals, distributing oxygen and nutrients to active tissues more rapidly. This elevated metabolic rate comes with costs. Gregarious locusts consume resources faster, age faster at the cellular level, and are more susceptible to the cascading consequences of energy deficit if food becomes unavailable during flight. The body that gregarization builds is one optimized for performance over longevity, a biological sprint configuration applied to an animal that may need to travel thousands of kilometers. It is a trade-off that makes complete sense for a creature whose survival depends not on living long but on moving far and consuming fast. Not all of the physical transformations of gregarization are as permanent as the neural changes described earlier. Some morphological features, particularly those encoded in the cuticle and expressed during molting, remain sensitive to environmental conditions through the nymphal stages. A nymph that has begun showing gregarious coloration yellowing, early melanin deposition, but has not yet completed its next molt can, if isolated and maintained in the chemical environment of solitary individuals, partially reverse those color changes in the subsequent cuticle. The reversal is rarely complete, intermediate colorations are common in partially reversed individuals, carrying a mosaic of solitary and gregarious pigment expression that reflects the mixed chemical signals received during the sensitive period before and during the molt. Wing elongation and muscle mass changes, which require tissue growth across the molt boundary, are somewhat less reversible than surface coloration. Once wing buds have committed to gregarious proportions, subsequent isolation cannot fully redirect the developmental trajectory. The brain changes described earlier are the most persistent. The window during which isolation can meaningfully alter the physical trajectory of a gregarizing locust is real, but narrow a few days in young nymphs, shorter in later instars, and effectively closed in adults. The transformation, like most developmental processes, is far easier to redirect early than to reverse late. Standing back from the accumulated detail of the gregarious transformation, the color shift, the wing elongation, the muscle reconfiguration, the brain expansion, the immune recalibration, the metabolic acceleration, what emerges is something 5 and 5 that resists easy summary but demands one anyway. The same genome, carrying the same nucleotide sequence in every cell, has produced two animals so different that trained entomologists once classified them as separate species. One is green, cautious, selective, slow, chemically invisible, and ecologically negligible. The other is yellow-black, hyperactive, broadly destructive, physically robust, neurologically expanded, and capable of participating in events that reshape the food security of entire regions. The mechanism that separates them is not mutation, not evolution in real time, not the accumulation of genetic difference across generations. It is gene expression, the selective activation and silencing of the same underlying instructions in response to the density of conspecifics in the environment. The locust has not changed its code. It has changed which parts of the code it reads, and in doing so, it has become by nearly every functional measure a different organism. What the locust demonstrates quietly without drama, simply by existing in two phases, is that identity is not stored in DNA alone. It is stored in the relationship between DNA and the world that reads it. There is no locust at the front of a swarm giving directions. There is no hierarchy, no chain of command, no individual whose decisions propagate outward to the rest. A swarm of several billion locusts achieves coherent collective navigation through a mechanism that has no center and no authority, a property that researchers call self-organization and that remains, even after decades of study, genuinely difficult to hold in the mind all at once. Each individual locust tracks a small number of nearest neighbors, those within its immediate visual and chemical range, and adjusts its own speed and direction in response to theirs. It moves when they move. It turns when the local average turns. It slows when those immediately around it slow. No single individual has information about the swarm's overall shape, size, or direction. Each one knows only its immediate neighborhood. And yet from this purely local information processing replicated simultaneously across billions of individuals, a coherent, directed, navigating collective emerges. The swarm moves. It turns. It avoids obstacles. It crosses terrain. It does all of this without any individual possessing the information required to do it alone. The mathematics underlying this process belong to a branch of physics concerned with the behavior of large numbers of interacting particles, and the locust swarm in formal models behaves with a regularity that those models predict well, which is its own kind of astonishment that billions of living animals, each following a handful of simple rules, produce behavior orderly enough to be described by equations. The precise number of neighbors a locust actively tracks has been studied through high-speed video analysis of swarm edges and laboratory group experiments. The evidence suggests that each individual maintains meaningful behavioral response to approximately 6 to 8 neighbors, a number strikingly similar to the topological neighbor count found in starling murmurations and some schooling fish species. This convergence across vastly different taxa suggests something important, that 6 to 8 neighbors may represent a near optimal number for balancing local responsiveness with global coherence in a self-organizing group. Too few tracked neighbors, and the group fragments local perturbations propagate only a short distance before dissipating. Too many, and the system becomes computationally demanding and sluggish. responding too slowly to local changes. 6 to 8 sits at a functional sweet spot. The locust does not know this. It is not optimizing. It is simply responding to what it can see and sense, and the sensory range of its compound eye and antennae happens to encompass roughly that number of individuals at typical swarm density. Evolution has produced a tracking range that generates coherent collective behavior, not through foresight, but through the elimination of individuals whose tracking range produced less coherent and therefore less survivable group dynamics across millions of generations of swarm life. The emergence of coherent group behavior from simple local rules is one of the central phenomena of complexity science, and the locust swarm is one of its most dramatic natural illustrations. When each of billions of individuals applies the same small set of rules, match the speed of your neighbors, align with their direction to maintain a minimum distance, the collective outcome is a structure with properties that no individual possesses or could generate alone. The swarm has a leading edge and a trailing population. It has internal density gradients. It responds to landscape features as a unit. It accelerates into favorable winds and slows in adverse ones. It navigates around mountain ranges and crosses water bodies by exploiting altitude and thermal lift. None of these are properties of any individual locust. They are properties of the interaction system features that exist only at the level of the collective and vanish the moment the collective disperses. Researchers studying complex systems describe this as emergent behavior, a term that is technically precise but risks sounding mystical. There is nothing mystical in it. There is, however, something that warrants genuine intellectual pause: the recognition that large numbers of simple interactions can produce outcomes of a complexity and coherence that the components themselves are entirely incapable of. The swarm is smarter than any locust in it, and no locust in it is responsible for that. Within the dense chemical environment of a moving swarm, pheromones serve a coordination function that operates in parallel with visual and mechanical neighbor tracking. The gregarious pheromone blend produced from the cuticle feces and respiratory secretions of individual locusts creates a distributed chemical field that carries information about local density, movement, direction, in some contexts, alarm. When the leading edge of a swarm encounters a food source, the feeding activity produces elevated pheromone concentrations in that zone that attract following individuals, a chemical beacon that reinforces the leading edge's position and draws the swarm's mass toward productive areas. When a section of the swarm is disturbed by a predator or an environmental obstacle, alarm pheromones released by disturbed individuals propagate through the local chemical field, triggering avoidance responses in neighbors who have not yet encountered the disturbance directly. The chemical field produced by a large swarm is, in effect, a distributed information network, a medium through which local events are broadcast to surrounding individuals without any direct contact between the source and the recipients. It is not language. It carries no symbolic content and no deliberate intent, but it moves information through a crowd of billions with a speed and reach that no individual signal alone could achieve, coordinating responses to events that most individuals will never directly perceive. The polarized light detection capability described in the solitary locust's compound eye finds its full expression in the swarm. During sustained migratory flight, crossing deserts, mountain ranges, and open water, the swarm maintains directional coherence over distances of hundreds of kilometers in conditions where ground-based landmarks are absent or irrelevant. The celestial polarization pattern, stable and predictable across the entire sky dome, provides a navigational reference that persists through partial cloud cover across all terrain types and at all altitudes accessible to locust flight. Individual locusts sample this pattern continuously during flight, using it to maintain a consistent body orientation relative to a celestial reference direction. When millions of individuals do this simultaneously, the result is a swarm with a collective heading that is remarkably stable over time, drifting gradually in response to the sun's movement across the sky as a compass needle drifts with the magnetic field, but maintaining directional integrity across the hours and distances of a migratory flight. Scientists working on autonomous navigation systems for aerial drones have studied the locust's polarized light compass with considerable interest, not because the locust system is the most sophisticated navigation tool in nature, but because it achieves long-range directional stability with minimal computational overhead. It is an elegant solution to a hard problem carried in the eye of an insect deployed at continental scale. The crossing of open water bodies by locust swarms is one of the more physically remarkable feats in animal migration. Locusts cannot swim. They cannot float for long. An individual locust that descends to sea level over open ocean is a dead locust. And yet swarms have been documented crossing the Red Sea, the Arabian Sea, and portions of the Atlantic Ocean distances up to several hundred kilometers of open water. The mechanism relies almost entirely on atmospheric physics. Swarms climb to altitude, sometimes several kilometers, where prevailing winds are faster and more consistently directional than at the surface. At these altitudes, the locusts are carried by the wind rather than flying against it, reducing the energetic cost of crossing to a fraction of what sustained active flight would require. Thermal columns, rising warm air masses produced by solar heating of the surface, provide lift that allows altitude maintenance with minimal wing effort. The swarm, in effect, becomes a passenger of the atmosphere rather than a vehicle moving through it. Individual locusts do lose altitude over extended crossings, and mortality during ocean crossings can be substantial. But the swarm's scale means that even significant individual mortality leaves millions of survivors to make landfall. On the far side, the ocean, which should be a barrier, becomes a corridor. The crossing is not a triumph of locust determination. It is a consequence of the locust's body weight, wing area, and the willingness of the atmosphere to carry things that make themselves available to be carried. A locust swarm is not a uniform cloud of individuals distributed evenly through space. It has internal architecture regions of higher and lower density functional zones that play different roles in the swarm's collective behavior and structural features that persist as the swarm moves and feeds. The leading edge of a mature swarm is typically the most dense and most behaviorally active zone. Individuals here are the first to encounter fresh vegetation, the first to land and begin feeding, and the primary source of the feeding pheromones that signal the rest of the swarm toward productive areas. Behind the leading edge, density drops and then rises again in the main swarm body, a dense, coherent mass where individual flight paths are tightly coordinated by neighbor interaction. The trailing population is sparser and more variable in composition, containing individuals that have fed recently and are flying more slowly. Individuals that have been displaced from the main body by turbulence, and in some swarms newly airborne individuals joining from egg hatching sites below. This internal structure is not designed or maintained by any coordinating mechanism. It is a natural consequence of the local interaction rules applied by billions of individuals simultaneously, structure that arises from behavior rather than being imposed upon it. The swarm organizes itself the way a river organizes its currents, not because any water molecule knows where the river is going, but because the physical rules governing each molecule's movement applied at scale to produce a coherent flow. A locust swarm, encountering a mountain range, a city, or a large body of standing water, does not plan a route around it. No individual perceives the obstacle in its totality. Each locust, flying at the scale of its own body, sees only the immediate visual field around it, the wing of a neighbor, the approaching surface of the ground, the thermal gradient of rising air. And yet swarms navigate around large obstacles with a coherence that, observed from altitude, resembles deliberate route planning. The mechanism is the same local interaction system that generates all swarm behavior applied to a new context. Individuals at the swarm's leading edge that encounter an obstacle, a cliff face, a building, an updraft produced by terrain, adjust their flight path away from it. Those immediately behind them, tracking their movement, adjust in the same direction. The adjustment propagates backward through the swarm in a wave of behavioral change that redirects the collective flow without any individual perceiving the reason for the change. The swarm flows around the obstacle the way water flows around a stone, not because the water knows the stone is there, but because the physical interaction between water and stone surface propagated through the fluid redirects the flow. The locust swarm is, in this mechanical sense, a fluid one made of living animals, each following its neighbors, producing a collective that moves with the logic of water and the appetite of something else entirely. There is a sound that accompanies a large locust swarm, one that people who have experienced it describe with a consistency that suggests the experience leaves a lasting impression. At a distance of several kilometers, a large flying swarm produces a low, continuous rushing sound, something between the noise of a strong wind through dense foliage and the distant approach of heavy rain on a dry surface. The sound is produced primarily by the combined wing beats of millions of individuals. Each locust's wings stroke at roughly 18 to 20 times per second, and the combined acoustic output of billions of simultaneous strokes produces a broadband noise that carries across open terrain. Closer in, the character of the sound changes. The rushing deepens and becomes more textured, layered with the individual stridulatory signals of millions of locusts rasping simultaneously, a dry, complex overlay on the fundamental wing beat frequency. Inside the swarm, observers describe the sound as immersive and disorienting, a sound that seems to come from everywhere simultaneously, that has no clear source and no clear edge, that fills the auditory field completely. People who have stood in the path of an approaching swarm and heard that sound growing from a distant suggestion to an encompassing presence describe something that bypasses analysis entirely. A sound that the body responds to before their mind has finished identifying what it is. A locust swarm is not merely moved by atmospheric conditions, it actively exploits them to manage the thermal environment of its constituent individuals. During the heat of the day in desert and semi-arid regions, surface temperatures can exceed the thermal tolerance limits of individual locusts flying close to the ground. Swarms respond to this by ascending to altitudes where ambient temperature is lower and the combined heat load from solar radiation and ground surface radiation is reduced. This altitude adjustment is not coordinated by any central signal. It emerges from the independent thermoregulatory decisions of millions of individuals, each responding to its own thermal discomfort by climbing, and the aggregate of those individual climbing decisions producing a collective altitude increase across the swarm. The same dynamic operates in reverse in the early morning when swarms descend from cooler altitudes as surface temperatures warm toward the optimal flight range. This collective thermoregulation gives the swarm a functional temperature envelope, a range of atmospheric conditions within which it can sustain flight that is broader than what any individual could maintain through behavioral thermoregulation alone, simply because the collective can access a wider range of altitudes and microclimates through its distributed self-organizing response. There is something quietly remarkable in the image of a billion animals, each adjusting its altitude by a few meters in response to its own skin temperature, and in doing so, keeping the entire collective within a thermal window that allows it to continue moving, none of them knowing the others are doing the same thing, all of them producing together what none could achieve alone. Aiding edge of a feeding swarm operates with a biological efficiency that agricultural scientists have measured carefully, and that still produces numbers difficult to absorb. As the front descends onto vegetation, individual locusts land orient to the nearest plant material and begin feeding within seconds. The feeding is not random. Locusts preferentially target actively growing tissue, particularly young leaves and reproductive structures, which carry the highest concentrations of protein and soluble carbohydrates. Stems and older leaf tissue are consumed only when more nutritious options are exhausted. The leading edge moves across the vegetation like a wave, with individuals feeding taking flight, moving forward as the immediately available material is depleted, and landing again on the next available plant. Behind this advancing front, the main swarm body follows, gleaning what the leading edge has left. By the time the trailing population passes through, the vegetation has typically been reduced to bare stems and soil. The efficiency of the leading edge's initial pass is high enough that in dense crops, maize, sorghum, millet, meaningful yield loss begins within minutes of the leading edge's arrival. Farmers who have watched a swarm's feeding front move across a field describe the experience with a specific kind of helplessness. The knowledge that what they are watching cannot be stopped, only witnessed. The consumption capacity of a large locust swarm resists intuition until it is anchored in something concrete. A single square kilometer of a dense swarm contains roughly 40 to 80 million locusts. Each adult locust consumes approximately its own body weight in vegetation daily, roughly 2 grams of fresh plant material. A swarm of 1 square kilometer therefore consumes between 80 and 160 metric tons of vegetation per day from that single square kilometer alone. Large swarms extend across hundreds of square kilometers. The 2020 East Africa swarms, among the largest documented in recent decades, covered areas exceeding 2,400 square kilometers at their peak. The total daily consumption of such a swarm reaches figures comparable to the daily caloric food requirement of approximately 35,000 people. Not as a rough estimate, but as a figure derived from the measured consumption rates of individual locusts scaled to documented swarm sizes. To place that alongside something a person can hold, a single day of feeding by a large swarm can consume the equivalent of what a mid-sized American city eats in a day, not its luxury food, not its waste, but its basic caloric sustenance stripped from the land in hours, leaving behind soil and bare stems and the farmers who planted the fields, standing at their edges. The timeline of crop destruction during a swarm's passage is measured not in days but in minutes to hours, depending on swarm density and crop type. High-value crops with dense canopy structure, mature maize millet at grain-fill stage vegetable gardens, can be reduced from full productivity to bare stalks within a single feeding event, lasting 2 to 4 hours. Cereal crops at earlier growth stages, where the vegetation is lower and less structurally complex, are stripped faster. Pasture grasses and rangeland vegetation, which lack the structural complexity that might slow individual feeding, are sometimes cleared to soil level in a single swarm passage. The destruction is not merely quantitative, it is also qualitative. Even plants that survive a swarm passage with some structural integrity remaining have typically lost their reproductive and photosynthetically active tissue, leaving them unable to recover productively within the growing season. Perennial plants with established root systems may regrow given adequate subsequent rainfall. Annual crops, which represent the majority of food production in the regions most vulnerable to locust activity, do not. The field that supported a swarm's feeding for one afternoon supports nothing that season. The loss is immediate, visible, and for subsistence farmers with no reserve stocks and no crop insurance, it is not merely agricultural. It is the difference between eating and not eating through the months that follow. The ecological consequences of a large swarm extend well beyond the immediate destruction of vegetation. When millions of locusts land, feed, defecate, and eventually die in a concentrated area, they deposit an enormous quantity of organic material into the soil and local water bodies. Locust feces, rich in nitrogen and partly digested plant compounds, can temporarily alter soil chemistry in areas of dense roosting, raising nitrogen levels sharply in the short term, but also introducing compounds that affect soil microbial communities in ways that take years to fully resolve. Water bodies near roosting sites receive both fecal input, and after the swarm moves on, the bodies of dead or dying individuals in quantities that can temporarily deplete dissolved oxygen and stress aquatic communities. The same dead locusts that contaminate water bodies, however, represent an enormous pulse of protein-rich biomass that supports rapid population increases in scavenging and predator species. Bird populations near swarm zones show dramatic short-term increases. Jackal, hyena, and various rodent populations exploit the carcass resource. Even some plant communities benefit from the nitrogen pulse in the medium term. The swarm is not only destructive, it is also, in a narrow ecological sense, generative, a massive concentrated transfer of energy from one part of the ecosystem to many others, violent in its mechanism, complex in its downstream effects. One of the more startling discoveries in locust swarm dynamics is that the collective forward movement of a marching or flying swarm is partly driven by a mechanism that has nothing to do with navigation or pheromone following. It is driven by hunger, specifically by the hunger of individuals at the rear of the swarm for the individuals immediately ahead of them. Locusts are not obligate cannibals, but under conditions of high density and insufficient vegetation, they will consume other locusts. A locust at the rear of a marching band, finding itself in territory already depleted by those ahead, faces a choice between stopping and being consumed by those behind or continuing to move forward. The mathematics of this pressure applied simultaneously to every individual in the trailing population, creates a persistent forward push that supplements the navigational and pheromone-based mechanisms driving swarm movement. Researchers have described this as a forced march dynamic, not metaphorically, but as a precise description of the mechanism. The swarm moves in part because stopping is dangerous. Each individual's forward movement is simultaneously a response to the resource depletion created by those ahead and the predation pressure created by those behind. The swarm consumes the landscape before it, partly to stay ahead of itself. The cannibalism dynamic described above has been studied in controlled laboratory conditions with a precision that leaves little ambiguity about the mechanism. When marching bands are prevented from moving forward, blocked by a barrier, the rate of cannibalistic attacks within the group increases sharply within hours. When the barrier is removed, forward movement resumes and attack rates decline. The relationship between movement opportunity and cannibalism rate has been replicated consistently enough to be considered established. What makes this finding particularly interesting is that it inverts the intuitive understanding of swarm movement as purely directed toward food. The swarm is moving toward food, yes, but it is also moving away from the locust behind it. The individual at the leading edge is the most fortunate, fresh vegetation ahead, no hungry neighbors in pursuit. The individual in the trailing population has depleted vegetation behind it, hungry neighbors immediately following, and only the forward movement of those ahead to follow. The swarm is in this sense both predator and prey, simultaneously a self-chasing system in which the pressure of pursuit and the promise of food ahead produce the same behavioral output: keep moving. Scientists still debate the relative contribution of cannibalism pressure versus resource seeking in driving swarm velocity under different field conditions, but few now doubt that both are real and that neither alone fully explains what is observed. The chemical environment left behind by a moving swarm is not cleared the moment the swarm passes. Volatile pheromone compounds released by millions of locusts accumulate in the air and on vegetation surfaces at concentrations that persist for hours after the swarm has moved on. This chemical persistence creates what researchers describe as a pheromone corridor, a lingering chemical trail that marks the swarm's recent passage and continues to attract locusts toward it even after the swarm itself has left the area. Stragglers, individuals separated from the main body by turbulence or obstruction, follow these chemical corridors to rejoin the collective. Smaller satellite populations, groups that have not yet fully joined a large swarm, are drawn toward the chemical signature of the main body's passage and converge on it over the hours and days following. The persistence of these chemical trails also means that the areas through which a large swarm has recently passed carry elevated locust-attracting chemistry in the soil and on vegetation surfaces, contributing to the tendency of swarms to move through the same geographic corridors in successive years, following routes that previous swarms have chemically marked. The landscape remembers the swarm, and the swarm in subsequent seasons finds those memories and follows them. A large locust swarm and the weather system above it are not independent phenomena, they interact. Swarms preferentially move with prevailing winds, exploiting airflow to extend their range with minimal energetic expenditure. The intertropical convergence zone, the band of low-pressure and converging trade winds that circles the globe near the equator and shifts seasonally, has a particularly significant relationship with desert locust swarm movement. Swarms in East Africa and the Arabian Peninsula track the seasonal position of this convergence zone with a fidelity that is not coincidental. The zone's convergence brings together wind flows from multiple directions, creating conditions that concentrate swarms just as drought concentrates populations. Researchers tracking swarm movements against atmospheric data have found that swarm trajectories align with wind patterns at the altitude of flight, with a correlation strong enough to serve as a predictive tool. Knowing where the winds are going is a reasonable first approximation of where the swarm will be tomorrow. The swarm does not read weather forecasts. A weather following system, one in which the aggregate of millions of individual decisions to fly with the wind rather than against it, produces a collective movement that mirrors the atmosphere's own large-scale organization. with a precision that took researchers some years to fully appreciate. The technology applied to locust monitoring has undergone a quiet revolution over the past 2 decades. Where earlier generations of locust researchers depended on ground-based scouts, aircraft observations, and retrospective mapping, from survivor reports, modern monitoring programs use a combination of satellite-based vegetation, indices, thermal imaging, and synthetic aperture radar to track swarm movement in near real time. Thermal imaging satellites detect the heat differential between a dense swarm and the surrounding environment. Millions of insect bodies at flight temperature produce a measurable thermal signature against a background of cooler atmosphere. This signature can be tracked frame by frame across successive satellite passes, generating movement trajectories and velocity estimates for large swarms without requiring any ground-based observer. Vegetation index data measuring the greenness of surface vegetation through spectral reflectance shows swarm passage as a sudden and geographically precise reduction in vegetation signal, allowing retrospective mapping of feeding areas and movement corridors. The combination of these data streams, integrated through dedicated monitoring platforms operated by the Food and Agriculture Organization of the United Nations, has substantially improved the lead time available to early warning systems. A swarm that would once have arrived at a vulnerable agricultural area with little or no warning can now, in favorable data conditions, be tracked for days before its arrival. Beyond tracking existing swarms, the current generation of locust forecasting tools attempts to predict where swarms will be before they arrive, a challenge that requires integrating atmospheric models, vegetation data, population density, estimates, and behavioral response curves into a single, predictive framework. Machine learning models trained on historical swarm movement data and atmospheric records have demonstrated the ability to predict swarm corridors, the geographic bands within which a swarm is likely to move over the following 72 hours with accuracy, substantially better than earlier rule-based forecasting approaches. These models learn the statistical relationships between atmospheric conditions, vegetation patterns, and swarm behavior from large historical datasets, and apply those relationships to current conditions to generate probabilistic forecasts. The 72-hour prediction window is not a hard limit but a practical one. Atmospheric forecast accuracy degrades beyond that horizon in ways that cascade through the swarm trajectory prediction, producing uncertainty ranges too wide for operational usefulness. Within 72 hours in favorable atmospheric data conditions, the models can identify likely movement corridors with enough spatial precision to guide pesticide deployment decisions and population evacuation planning in vulnerable agricultural areas. Scientists still debate the ceiling of predictive accuracy achievable with current approaches, whether the fundamental stochasticity of swarm behavior places a hard limit on forecast precision, or whether additional data streams and more sophisticated modeling could push useful prediction windows further out. The honest answer for now is that no one is certain. The historical record of locust swarms is long enough and geographically broad enough to constitute a recurring motif in human agricultural civilization, a pattern of catastrophic vegetation loss that has shaped food systems migration, patterns and political structures across multiple continents and millennia. The 1,875 outbreak of the Rocky Mountain locust across the American Great Plains remains, by some estimates, the largest documented insect aggregation in recorded history, a swarm estimated at 198,000 square miles that moved across Kansas, Nebraska, and adjacent territories, destroying crops across an area equivalent in size to California. The economic and human cost was severe enough to alter settlement patterns in the affected regions. The species itself, Melanopus spretus, was extinct within 3 decades, its breeding grounds in the mountain valleys of the Rocky Mountain states destroyed by agricultural development, making it the only major pest insect to have been eliminated by human land use. The 2020 East Africa crisis, by contrast, involved a species still very much present, the desert locust swarms that swept through Ethiopia, Kenya, Somalia, and neighboring countries, represented the worst outbreak in the region in 70 years, threatening the food security of tens of millions of people and arriving at the same time as a global pandemic that severely compromised the international response capacity. Two events, separated by 145 years on different continents involving different species, the same fundamental story. The largest desert locust swarms on record contain numbers of individuals that exceed the capacity of human intuition to process directly. Estimates for major swarms range from several billion to upward of 80 billion individuals, a figure derived from area measurements, aerial density surveys, and individual locust count per unit area. 80 billion is a number that requires translation to mean anything. If every person currently alive on Earth were assigned a portion of a swarm of 80 billion locusts, each person would be responsible for roughly 10 locusts. If those 80 billion locusts were laid end to end, each adult locust approximately 5 centimeters long, the resulting line would circle the Earth's equator more than 100 times. If they were stacked in a single column, that column would extend well beyond the moon. These translations are, in a sense, inadequate. They replace one incomprehensible number with a series of incomprehensible distances. But they gesture toward the correct intuition, which is that the scale of a major locust swarm exceeds the cognitive categories that human beings use to process large quantities. What is perhaps more useful than any numerical translation is this: a swarm of that size moving across a landscape is not an event that the landscape survives unchanged. It is a geological moment in biological time, something that reshapes the distribution of matter across hundreds of square kilometers in the course of a single day. The economic consequences of a major locust outbreak extend far beyond the immediate destruction of standing crops. The 2020 East Africa outbreak is estimated to have caused agricultural losses exceeding $3 billion across the affected region, a figure that encompasses crop destruction, livestock forage loss, emergency pesticide deployment costs, and the downstream economic effects of food price increases in affected markets. For subsistence farming households, which constitute the majority of the agricultural workforce in the most heavily affected areas, the economic impact is not denominated in dollars but in months without food and children withdrawn from school to help surviving family members replant, in livestock sold at distressed prices to cover immediate food needs. The cascading effects of a single major swarm event can persist across multiple agricultural seasons. The loss of seed stock, the depletion of household financial reserves, and the physical and psychological toll of repeated crop failure combine to set household economic trajectories back by years. Food security analysts treat major locust outbreaks as category-level shocks events comparable in their economic impact on vulnerable agricultural populations to severe droughts or regional conflicts, and the international response frameworks developed for them reflect that categorization. A swarm is not merely an agricultural problem, it is a humanitarian event denominated in hunger, wearing the shape of an insect. A sufficiently large, dense locust swarm does not merely move through the atmosphere, it modifies it locally. The combined water vapor released through the respiration and cuticle transpiration of billions of individuals adds measurable humidity to the air within and immediately below a dense swarm. The wing beats of billions of locusts produce mechanical turbulence at the scale of the swarm's underside, a boundary layer disruption that alters the wind profile close to the ground surface below the swarm, affecting the microclimate experienced by the vegetation being consumed. Dense swarms reduce incoming solar radiation to the surface beneath them, not dramatically but measurably, in the same way that a thin cloud layer reduces light intensity. This reduction in insolation can temporarily lower surface temperatures under the swarm, creating a microclimate that differs measurably from surrounding unshaded areas. Whether any of these micro-weather effects influence locust behavior directly, whether the humidity, turbulence, or light reduction produced by the swarm feeds back into individual locust decisions about altitude, direction, or feeding, is a question researchers have begun examining but not yet fully answered. The possibility that a swarm large enough to modify local atmospheric conditions might then respond to those modifications is a feedback loop that, if confirmed, would add another layer to what is already a deeply self-organizing system. The same properties that make a locust swarm overwhelming at its peak also render it vulnerable in ways that are not immediately obvious from the outside. The swarm's power is inseparable from its density, and density requires the continuous co-location of billions of individuals in a shared atmospheric and chemical environment. That shared environment is a vector for the same entomopathogenic fungi and parasitic organisms described in the solitary phase now operating at swarm scale. A fungal epizootic, an outbreak of fungal disease moving through a dense population, can cause catastrophic locust mortality within a swarm in days. The Metahizium acridum fungus, a species highly specific to locusts and grasshoppers, has been developed as a biopesticide precisely because of its capacity to spread through dense locust populations. The swarm's density also creates vulnerability to targeted chemical intervention. A pesticide application that encounters a resting swarm at high density achieves a contact rate impossible against a dispersed population. Weather is another exploitable fragility. Cold fronts that ground the swarm, rain events that weigh down wings and create fungal conditions, prolonged headwinds that exhaust fat reserves before food sources are reached. The swarm that seems from the outside like an impregnable force, and in many respects carries within it the same biological vulnerabilities as every living system. It is powerful, and it is mortal. It dominates landscapes and it dies in the rain. That tension between overwhelming collective capability and the fragility of every individual within the collective is perhaps the most honest summary of what the swarm actually is. The eighth plague of Egypt, described in the Book of Exodus as a locust swarm so dense it darkened the sky and consumed every plant the preceding hail had left standing, is not merely a theological narrative. It has an ecological one. Researchers examining the sequence of events described in Exodus against the known climatology and locust biology of the Nile Delta region have found a coherent natural chain. The flooding and agricultural disruption of preceding plagues would have produced precisely the concentrated vegetation and elevated soil moisture conditions that support rapid locust population growth and gregarization. The prevailing wind patterns of the eastern Mediterranean, specifically the Khamsin, the hot southeasterly wind that brings locusts from the breeding grounds of the Arabian Peninsula and East Africa into Egypt, align with the plague narrative's description of an east wind carrying the swarm in. The subsequent departure of the swarm, driven away by a strong west wind according to the text, matches the dispersal mechanism for swarms that have exhausted their food supply. The Exodus account does not require miraculous mechanics to be ecologically consistent. It requires a bad year in East Africa, a good monsoon that concentrated populations, a Khamsin that arrived on schedule, and a civilization organized enough to record what happened when those factors converged on the Nile Delta simultaneously. The miracle, if one is inclined to find one, may lie less in the swarm's arrival than in what its departure left behind. The locust swarm is one of the few natural phenomena to appear independently as a major narrative element across the foundational texts of multiple distinct civilizations and religious traditions. The Book of Joel, one of the Hebrew prophetic texts, opens with a locust invasion of such severity that it serves as the central metaphor for divine judgment, an event so total in its destruction that the prophet demands it be told to children and grandchildren as a permanent cultural memory. The Quran references locusts among the signs sent to Pharaoh, embedding them in the same narrative tradition as the Exodus account, but within a distinct theological framework. The Mahabharata, the Sanskrit epic of the Indian subcontinent, contains passages describing locust swarms as omens of war and civilizational collapse, associating their appearance with political catastrophe in ways that suggest the Indian subcontinent's own long history of devastating outbreaks. These independent appearances in texts, separated by geography, language, theology, and centuries, suggest something important: that the locust swarm made an impression on human civilizations deep enough to be preserved not as agricultural record but as mythology, elevated from natural disaster to cosmic significance, written into the foundational stories that cultures tell about the nature of power, punishment, and human vulnerability. The locust did not earn its place in Scripture by being large. It earned it by being in the memory of the people who witnessed it total. The extinction of the Rocky Mountain locust, Metamorphosis is one of the more sobering episodes in the intersection of entomology and environmental history. The species was in the 1870s the most economically destructive insect in North America. The 1,875 swarm alone was estimated to cover an area larger than California and to contain more individual insects than any other documented aggregation in the continent's recorded history. 30 years later, the species was gone. The mechanism of its extinction was not deliberate. Settlers moving into the river valleys of Montana, Wyoming, and adjacent territories plowed, irrigated, and cultivated the moist, loamy soils of those valleys. The specific habitat in which the Rocky Mountain locust laid its eggs and maintained its permanent breeding populations. As that habitat was converted to agriculture, the breeding population shrank. The migratory swarms, which had seemed inexhaustible, were drawing on a reservoir that was being emptied from below. By 1902, the last confirmed living specimens were recorded. Preserved egg pods have been recovered from glacial ice in Montana, allowing genetic analysis of the species, but not its return. The lesson the Rocky Mountain locust offers is an uncomfortable one: that a species capable of producing the largest insect swarm ever recorded can be eliminated not by any targeted effort, but simply by the conversion of a relatively small area of specific habitat without anyone noticing until the swarms stop coming. The desert locust's relationship with time is not limited to the lifespan of any individual. Its eggs, buried in the soil by gregarious females during outbreak years, can enter a state of developmental arrest, diapause, that suspends their development almost indefinitely under conditions of heat and desiccation. In this arrested state, the embryo inside the egg maintains minimal metabolic activity, consuming its yolk reserves at rates slow enough to sustain viability across months, and in some documented cases years of dormant waiting. The trigger for resumed development is moisture when rainfall sufficient to percolate to the egg's burial depth arrives. The dormant embryo resumes development and progresses toward hatching within days to weeks. This dormancy mechanism means that the consequence of a major locust outbreak is not limited to the season in which it occurs. The eggs laid by millions of gregarious females during an outbreak year persist in the soil as a latent population, a biological time capsule of the outbreak's density, waiting for the next rainfall event that will activate it. An area that experienced an outbreak 3 years ago and has shown no visible locust activity since may harbor in its soil a population capable of producing another outbreak the moment conditions become favorable. The land remembers in this sense in a way that is entirely literal. The memory is stored underground, in the arrested embryos of the last generation. The structural resilience of locust egg pods is a product of evolutionary pressure applied across millions of years of existence in environments where soil conditions are violent and unpredictable. Each egg pod is a sealed unit, a cluster of 60 to 160 eggs arranged in a column within a hardened foam matrix, enclosed by a plug of the same material at the soil surface. The foam matrix is not merely structural padding. It is a hygroscopic material that manages moisture exchange between the eggs and the surrounding soil, buffering the eggs against both desiccation and waterlogging by absorbing and releasing moisture slowly. Laboratory tests of egg pod resilience have subjected them to temperatures exceeding 50 degrees Celsius for extended periods, to soil moisture conditions ranging from severe drought to brief flooding, and to physical compression from overlying soil pressure, and found that hatching success remains measurable across a surprisingly wide range of these stresses. The eggs are not indestructible, but they are substantially more tolerant of environmental extremes than the adult locusts that produced them. This resilience means that control efforts aimed at adult swarms, even highly effective ones, do not necessarily interrupt the next generation's emergence. If egg pods laid before the control effort, hatching conditions are eventually met. Controlling a locust outbreak and preventing the next one are for this reason two entirely different problems. The challenge of locating locust egg pods buried in soil, invisible from the surface, variable in their precise location, and distributed across enormous areas of semi-arid terrain. has driven the development of detection technologies borrowed from archaeological and geological survey practice. Ground-penetrating radar systems, adapted for the specific depth range and target size of locust egg pods, have been tested in field conditions across East Africa and the Arabian Peninsula. The radar emits short pulses of electromagnetic energy into the soil and measures the reflections produced by density discontinuities, boundaries between materials of different composition and moisture content. An egg pod buried in sandy soil produces a reflection pattern distinguishable from background soil structure, allowing its detection without excavation. The technology is not yet operational at the scale needed for routine monitoring. The equipment remains expensive, the data processing demanding, and the false positive rate under heterogeneous soil conditions still too high for reliable operational use. But proof-of-concept studies have demonstrated that egg pod detection from the surface is physically achievable, and the development trajectory of the technology suggests that operational ground-penetrating radar surveys for locust egg mapping may become feasible within the coming decade. When that capability is realized, it will represent a fundamental shift in the temporal horizon of locust management, from responding to outbreaks after they have already begun, to identifying and addressing the conditions for the next one while it is still underground, still waiting, still dormant in the dark. The international infrastructure now dedicated to locust monitoring represents one of the more extensive applications of remote sensing and machine learning to a single biological threat. The Food and Agriculture Organization's Desert Locust Information Service operates a continuous monitoring system, integrating data from multiple satellite platforms, optical imaging satellites for vegetation and surface condition mapping, meteorological satellites for wind and rainfall tracking, and synthetic aperture radar satellites for detecting large roosting concentrations. These data streams are processed through machine learning models trained to identify locust-favorable conditions, flag anomalous vegetation loss patterns consistent with swarm feeding, and generate probabilistic outbreak risk maps updated on daily to weekly cycles. Ground truth for these models comes from a network of national locust monitoring units across the 30+ countries within the desert locust's potential range. Field observers who report population observations, development stages, and behavior through a standardized data collection system that feeds directly into the central modeling framework. The system is imperfect and resource-constrained. Several of the countries within the locust range have monitoring capacity that falls well short of what the models require for reliable local prediction. But the architecture that exists represents a genuine achievement. A planet-scale early warning system built around the life cycle of a 2-gram insect, drawing on technology that includes machine learning, satellite radar, and the field notebooks of observers walking through East African scrubland at dawn. The chemical pesticides used in conventional locust control, primarily organophosphate compounds, applied by aircraft over large areas, are effective against adult swarms and nymphal bands but carry substantial non-target ecological costs. They are broadly toxic to other insects, including pollinators and the natural enemies of other agricultural pests, and their application over the semi-arid ecosystems where locusts breed can produce cascading ecological effects that persist well beyond the outbreak event. Biological control programs offer an alternative that trades speed for specificity. The Metahismium acridum biopesticide, derived from the locust-specific strain of the entomopathogenic fungus described earlier, kills locusts with high specificity and limited impact on non-target species. Applied as a spray of fungal spores, it spreads through contact among locusts in the target population, producing mortality across a timescale of 7 to 14 days rather than hours. This slower action makes it less effective for emergency response against fast-moving adult swarms threatening imminent crop destruction. It is more suitable for application against nymphal bands in the early stages of swarm formation, where the slower kill timeline is operationally acceptable, and the opportunity to prevent swarm development justifies the trade-off. Parasitic wasps, tachinid flies, and avian predators are also being examined as components of integrated management approaches. Systems that combine biological, chemical, and cultural control methods in sequences timed to the locust's developmental stage and the operational context of the response. The same animal that empties fields and threatens food security for millions is itself a food source for approximately 2 billion people across Africa, Asia, and the Middle East. A nutritional relationship that predates agriculture and has persisted through every intervening century. Locusts are consumed across their range in a variety of preparations: roasted, fried, dried and ground into flour, boiled in salt water, or eaten raw immediately after capture. Their nutritional profile is genuinely impressive. Dried locust contains between 50 and 65% protein by dry weight, with a complete amino acid profile comparable to conventional livestock meat. Their fat content runs to approximately 15 to 20% of dry weight, dominated by unsaturated fatty acids. They carry meaningful quantities of iron, zinc, magnesium, and B vitamins, micronutrients whose deficiency drives significant disease burden in the populations where locust consumption is most common. The Food and Agriculture Organization has documented locust consumption as a sustainable protein source with substantially lower land, water, and greenhouse gas costs per unit of protein than conventional livestock. The irony that one of the most destructive agricultural pests in human history is also one of the most nutritionally efficient protein sources available to the people it most frequently affects is not lost on food security to researchers, who note with some care that the relationship between locusts and the human communities in their path has never been purely one of victim and disaster. It has also, across millennia, been one of predator and prey. The desert locust begins its life as an exercise in restraint, green, careful, alone, an animal whose entire existence is organized around the avoidance of notice, the maintenance of distance, and the quiet negotiation of a difficult landscape. It feeds with precision. It navigates with elegance. It touches nothing it does not need and asks for nothing from the world beyond what it can find without disturbing anything around it. It is by nearly every measure a model of biological efficiency in solitude, and then someone touches its leg. Again and again and again, a molecule accumulates. A cascade begins. The same body carrying the same instructions in every cell begins building a different animal from the inside out. Different color, different wings, different brain, different appetite, different relationship to every living thing it encounters. The transformation does not require a new genome. It does not require a generation. It requires proximity and time and the chemical arithmetic of a nervous system counting touches and reporting upward when the count grows too high. What the locust teaches, quietly without apparent awareness of the lesson it is offering, is that the line between one kind of being and another can be thinner than intuition allows. That identity is not fixed in biology the way an address is fixed on a map. That the same fundamental instructions read in a different context can produce something so different from the original that careful observers once believed they were looking at two entirely separate species. Scientists study the locust because it destroys crops, threatens food systems, and crosses continents. Those are the reasons the funding exists and the satellites are in orbit. But the locust also offers something that no funding proposal has ever quite captured. A demonstration written in chemistry and behavior, and the bodies of billions of small animals moving across a continent. That identity is a relationship between an organism and its world, not a property the organism possesses alone. The solitary locust sitting still on its green stem at dawn, orienting its body toward the rising sun for warmth, navigating by the geometry of light, scattered across a sky it does not understand. That animal is not waiting to become something else. It has no intention of becoming something else. It is simply living the only life its current chemistry allows. The swarm is not its destiny. It is its response. And the distance between those two things, between what an organism is and what it becomes when the world presses close enough, is in the end what this story has always been about. And so we leave the desert locust where it began, on a stem in the quiet wearing green. Somewhere tonight, in the margins of East Africa or the Arabian Peninsula, a solitary individual is doing exactly what its biology asks, staying still, staying hidden, staying alone. It does not know what it could become. or that the distance between its current life and a swarm that darkens the sky is measured in the concentration of a single molecule in a structure smaller than a grain of sand. The most dramatic transformation in the animal kingdom begins in the most ordinary moment imaginable. Not with a storm, not with a signal, but with two small bodies moving through the dark close enough to touch. Thank you for spending this time with us at Secrets After Midnight. Good night.